Ini juga jumbo.
Assalamualaikum warahmatullahi wabarakatuh.
Good morning, ladies and gentlemen. A very warm greeting to each one of you. Welcome to the Department of Biology, Faculty of Mathematics and Natural Sciences, University of Jember, Indonesia.
Selamat pagi, Bapak dan Ibu. Salam hangat dan sejahtera untuk kita semua. Selamat datang di jurusan Biologi, Fakultas Matematika dan Ilmu Pengetahuan Alam, Universitas Jember, Indonesia.
My name is Salsabila Syafa'a and my colleague Muhammad Hilman Zahi. As the Master of Ceremony, we are very happy to welcome you in this opening ceremony of the 5th International Conference of Life Science and Biotechnology 2023.
Saya, Muhammad Hilman Zahi, dan rekan saya, Salsabila Syafa'ah, MC acara ini. Atas nama panitia The 5th International Conference on Life Science and Biotechnology 2023, kami sangat berbahagia menyambut Bapak Ibu semua dalam acara pembukaan konferensi ini.
Rector of the University of Jember, the Honorable Dr. Insinyur Iwan Taruna M.Eng, Dean of Faculty of Mathematics and Natural Sciences, Dr. Andes Ahmad Syaifullah MSc PhD, Head of Biology Department, Dr. Retno Wimbaningrum, Chairman of the Organizing Committee, Dr. Ernat Fuad Bahrul Ulum, SSI MSc. We will also to welcome and recognize the first day plenary speaker, Dr. Asmoro Lelono, Dr. Ernat Arif Lukman MT, and Professor Dr. Tirumalai Sami P. Philevan.
Yang terhormat, Rektor Universitas Jember, Dr. Insinyur Iwan Taruna M.Eng. Yang saya hormati, Dekan Fakultas MIPA, Bapak Dr. Andes Ahmad Syaifullah, MSi PhD, serta Ketua Jurusan Biologi, Dr. Retno Wimbaningrum, dan tidak lupa Ketua Panitia, Dr. Ernat Fuad Bahrul Ulum, SSI MSc. Tidak lupa, kami juga ingin mengucapkan selamat datang kepada pembicara pleno hari yang pertama, yaitu ada Bapak Dr. Asmoro Lelono, ada Bapak Dr. Ernat Arif Lukman, dan juga Profesor Dr. Tirumalai Sami P. Philevan. Honorable, distinguished speakers, participants, ladies and gentlemen. Let us praise and thank God the Almighty that we can gather here in a good condition. For your information, the theme of this 5th ICOLIB is Global Insight and Innovations of Life Sciences Research in the Post-COVID-19 Era. This year's meeting will provide you with plenty of opportunities to share and discuss knowledge, insight related to the topics.
Yang saya hormati Bapak Ibu pimpinan dan hadirin sekalian, marilah kita panjatkan puji syukur kehadirat Tuhan Yang Maha Esa atas limpahan karunia-Nya sehingga kita dapat berkumpul pada acara pagi hari ini dalam keadaan sehat walafiat. Perlu Bapak Ibu ketahui bahwa tema dari ICOLIB kelima ini adalah Global Insights and Innovations of Life Science Research in the Post-COVID-19 Era. Kami berharap dari acara hari ini, Bapak Ibu sekalian dapat berbagi ilmu pengetahuan, wawasan, serta berdiskusi tentang topik-topik tersebut.
Now, we would like to commence this event by singing the national anthem of the Republic Indonesia, Indonesia Raya, followed by the hymn of the University of Jember. Ladies and gentlemen, please rise for the national anthem.
Marilah kita awali acara pada pagi hari ini dengan menyanyikan lagu kebangsaan Indonesia, Indonesia Raya, dan akan dilanjutkan dengan himne Universitas Jember. Hadirin dimohon untuk berdiri.
Thank you very much. You may now be seated.
Terima kasih. Hadirin dipersilakan duduk kembali.
Ladies and gentlemen, now it's time for us to listen the first remarks which will be delivered by Dr. Renat Fuad Barul Ulum, MS, SSI, MSI, as the chairman of the organizing committee for the 5th ICOLIB. Please welcome Bapak Dr. Renat Fuad Barul Ulum, SSI, MSI.
Bapak dan Ibu hadirin sekalian, sekarang saatnya kita mendengarkan sambutan yang pertama yang akan disampaikan oleh Bapak Dr. Renat Fuad Bahrul Ulum, SSI MSC, selaku Ketua Penyelenggara ICOLIB kelima. Kepada Bapak Dr. Renat Fuad Bahrul Ulum, SSI MSC, dipersilahkan.
Honorable Rector, the University of Jember, Dean of the Faculty of Mathematics and Sciences, Head of Biology Department, distinguished guests, and the keynote speaker, dear colleagues and the ICOLIB Committee. Assalamualaikum warahmatullahi wabarakatuh. On behalf of organizing committee, I would like to welcome you all to the 4th International Conference of Life Sciences and Biotechnology, ICOLIB. The ICOLIB is a biannual international conference organized by Biology Department of the Faculty of Mathematics and Sciences, the University of Jember. The theme of the 4th conference is Global Insights and Innovation of Life Sciences Research in Post-COVID-19 Era. The scientific conference aims to create space for the presentation of current results of scientific work and for creating collaborative links between academics and professionals. This time, we have 6 invited speakers and a total of 134 oral presenters. The countries of origin of foreigners are from Germany, Pakistan, Argentina, India, and Philippines. The Indonesian participants are lecturers from Indonesian universities and researchers from the National Research and Innovation Agency, or BRIN. On the first day, we have 2 plenary sessions presented by Dr. Asmoro Lelono from University of Jember, Dr. Retner Alif Lukman from Julius Maximilian University of Werburg, Germany, and from Professor Tirumalai Sami P. Vilavan from University of Tübingen. On the second day, we will have 3 presenters by Professor Sunandar, PhD from Muhammadiyah University Purwokerto, and the second by Dr. Renat Abdul Hadiswandi from Medicine Hochschule Hannover, Germany, and the last by Dr. Diego Hojgaard from Leibniz Institute of Plant Sciences. In the parallel session, a total of 134 presenters spread over 18 rooms will present and discuss their scientific papers. The moderator of parallel session are mainly our alumni with an international study background. The 4th ICOLIB event had been published in BioWeb of Science, our Scopus partner of proceeding publication. A total maximum of 50 articles will be published in Scopus Indexes proceeding of BioWeb of Science, and other papers will be continuously peer-reviewed in Q4 journal IG Biotech, GID, Life Sciences and Biotechnology, the Biology Department Journal, and also ISSN proceeding of ICOLIB. To all members of organizing committee, the International Scientific Committee, the reviewers, and the collaboration partner, we would like to thank all of them for their tremendous effort to organize this conference successfully. We look forward to having a successful conference, and we hope that all attendees enjoy this conference. Thank you very much.
His Excellencies, distinguished speakers, participants, ladies and gentlemen. Now, we would like to request the honor from the Rector of the University of Jember, Dr. Insinyur Iwan Taruna M.Eng, to kindly present to the stage to deliver the speech and officially open the 5th International Conference on Life Science and Biotechnology 2023.
Bapak Ibu pimpinan yang saya hormati dan hadirin sekalian. Berikutnya kami mohon dengan hormat Rektor Universitas Jember Bapak Dr. Insinyur Iwan Taruna M.Eng untuk menyampaikan pidato pembukaan sekaligus secara resmi membuka the 5th International Conference on Life Science and Biotechnology 2023.
Please welcome the Rector of the University of Jember.
Hadirin sekalian, Rektor Universitas Jember.
Right, thank you for the opportunity to come to this conference, although by online. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to everyone, distinguished participants and speakers, my dear colleagues, ladies and gentlemen. Alhamdulillah, for the blessing and mercy of Allah Subhanahu wa Ta'ala, it's our God, we can be here in very good health so that we can join and participate in that we call it the 5th International Conference of Life Science and Biotechnology, ICOLIB 2023. On behalf of the University of Jember, of course, it is a pleasure and honor to welcome you all to this conference, which is organized by the Department of the Biology, Faculty of Mathematics and Natural Sciences, University of Jember. I got notice that this conference has reached about 134 papers. It indicate that this conference still attract the attention. From the participant, both whether from domestic or from the foreign country. Therefore, I would like to deliver my deepest gratitude to the chairman, Dr. Fuad Badrul Ulum, also with the team, and as well as to the Dean of the Faculty of Mathematics and Natural Sciences, Bapak Ahmad Saifullah, PhD. Yes, due to their hard work and valuable contribution to make to the reality. Also, I'd like to extend a special welcome and thanks to our, invited or prominent speaker who will be presenting and discussing the topic related to this conference theme. It is great honor for us during this conference to have Professor Velavan from University of Tübingen, Professor Sisunandar from Muhammadiyah University, Purwokerto. Also, there are also a speaker from our university. There are Dr. Asmarul Lono, then the other Dr. Abdul Hadi Swandi from Medicinist Hochschule Hannover, and Dr. Renat Arif Lukman from Julius Maximilian University of Würzburg, Germany. I hope to all the participants, you all enjoy the exciting speakers to come. Dear participants, ladies and gentlemen, I think this year conference theme could not be more pertinent, because in the past few years, have presented humanity with unprecedented trial, testing our resilience, adaptability, and scientific prowess. The pandemic brought our societies to a standstill. Revealing vulnerabilities in our healthcare system, and prompting us to rethink the way we approach life sciences research. So, our aim during this conference is to foster a deeper understanding of the challenges we face and the opportunities that lie ahead during our life. So, as the rector of the University of Jember, I expect that all the scientific paper produced during this conference shared during this conference will inspire us to be, more productive in this field, and also enable further improvement of our work. And yes, this is, we hope all, keeping us to stay at the leading edge of recent advances in life sciences. Moreover, I appreciate and fully support the holding of this conference. As I always mention, all the conference relevant to the university vision and mission can be, can be used, or we expect to be excellent, can be, empower our mission that we want to be a university that excellent in science, technology, and art, that with orientation in environmental sustainability, business, industrial agriculture. I think that's all from me. Again, I want to express my deepest gratitude again to the organizer, speakers, and participants who have made this conference possible. Let us seize the opportunity to learn from one another, to innovate, and to build brighter future for all. Well, by saying Bismillahirrahmanirrahim, let the 5th Conference on Global Insight and Innovation of Life Science Research in the post-COVID-19 era begin. I wish you all very productive conference with exciting and encouraging discussion and exchange of knowledge. Last but not the least, on behalf of the University of Jember, I'd like to sincerely apologize should there be any inconveniences that occur before, during, and after the event. Thank you. Ahlul kalam. Wassalamualaikum warahmatullahi wabarakatuh.
Ladies and gentlemen, the following agenda was words of prayer to have blessing from the God the Almighty for the success of this event. For this, we would like to invite to the stage Bapak Dr. Bambang Piluharto, SSI, MSI.
Hadirin sekalian, demi kelancaran dan kesuksesan acara ICOLIB kelima, marilah kita bersama-sama berdoa menurut agama dan kepercayaan masing-masing. Untuk itu, kami mengundang dengan hormat Bapak Dr. Bambang Piluharto, SSI, MSI, untuk memimpin doa I. Kepada Bapak Dr. Bambang Piluharto, dipersilakan.
Assalamualaikum warahmatullahi wabarakatuh. Before we start to our seminar, let's pray together to quote. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Hamdan syakirin, hamdan na'imin, hamdan yuafi ni'amahu wa yukafi'u mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karimi wa azimi sultanik. Praise be to you, oh my God. You are the source of salvation. You are of the source of happiness. Only to you all happiness will return. Thus revive ourselves with your happiness. Revive ourselves together those whom you gave happiness. And write us into heaven Darussalam, a paradise where happiness is. With your grace, oh my God, today we gather, grateful for the blessing that you have given to us, blessing of faith, blessing of heart, and blessing of knowledge. We are aware that you are the owner of blessing, no blessing on ourselves except for you, from you. Allahumma inni as'aluka ilman nafi'an, wa rizqan thayyiban, wa amalan mutaqabbalan. Rabbana dhallamna anfusana wa illam taghfir lana wa tarhamna lanakunanna minal khasirin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Assalamualaikum warahmatullahi wabarakatuh.
Now, as we've been officially starting the 5th ICOLIB, we would like to request all officials, distinguished speakers, and all participants to have a documentation session. We kindly remind you all to turn on your camera.
Bapak, Ibu, dan hadirin yang berbahagia, konferensi ICOLIB kelima telah secara resmi dimulai. Maka dengan hormat kami mohon kepada Bapak Ibu untuk berkenan memasuki acara selanjutnya, yaitu sesi dokumentasi. Mohon kiranya Bapak Ibu untuk menyalakan kamera. Dan untuk kepada pimpinan yang berada di ruangan dipersilahkan untuk maju ke depan untuk melakukan sesi dokumentasi. Terima kasih. Thank you very much. Now, we will present the video profile of the Biology Department, Faculty of Mathematics and Natural Sciences, University of Jember, Indonesia. The main organizer for this biannually event, International Conference on Life Science and Biotechnology. This event is organized by the support of the sponsor Nikon, Infinity, and Eppendorf. And we will also show you the video from the sponsors.
Hadirin sekalian, kami akan menyajikan video profil dari jurusan Biologi Fakultas MIPA Universitas Jember yang merupakan penyelenggara utama dari acara 2 tahunan International Conference on Life Science and Biotechnology. Acara ini didukung oleh sponsor Nikon, Infinity, dan Eppendorf. Kami juga akan menyajikan video dari sponsor acara ini.
The Biology Department of University of Jember has offered undergraduate studies since 2001 and has opened the master studies since 2010. The undergraduate study and master study both have accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of a management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Biology department Department of University of Jember, supported by well-equipped facilities. There are classrooms and laboratories giving students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and life certificate for tissue culture skills. The Biology Department of University of Jember provides the student central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research outreach for the community services, and student associations and extracurricular activities. The students and the lecturers have the opportunity to write a research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis sustainable.
Nikon's new digital medical device for live on-screen pathology imaging. Streamline your workflow. High-quality images captured quickly and easily. A real solution for digital pathology, ICOLIB UI. Observe images in real time. Place the sample with one hand. Wait just 2.5 seconds. Monitor observation for streamlining your workflow. It is no longer necessary to sit for hours looking through microscope eyepieces. The images are shared on screen, suitable for 2 or more people to discuss the samples. The macro image is always on screen to help navigate and find areas of specific interest. Nikon's proprietary imaging technology is also applied. Color reproducibility, after image reduction, and speedy scrolling. Nikon's legendary CF5 Plan Fluor series. Objective lenses are adopted for higher viewability. A real solution for digital pathology. ICOLIB UI.
Audio Jungle.
Can you be certain what the future holds? Neither can we. But we know from experience that time brings change and alter questions. Will our world have enough food? Is there a cure for dementia or cancer? What can we learn from the past? Will we take to the stars? And are we even asking the right questions? To find answers, we need development and innovation. And sometimes, the tiniest discoveries and achievements are the things that ultimately have the greatest impact. Still, we don't know what will come, but we know that you will find out. And we will do our best to ensure you can rely on us. Just like for the last 75 years, we are working every day to make your job more efficient. Because only then can we look towards a future that inspires and improves people's living conditions. Together.
And now we come to the end of the ceremony opening for the 5th ICOLIB. Thank you very much, ladies and gentlemen, for your participation in the opening ceremony. We do hope that you will have a fruitful and productive conference during this 2 days event.
Hadirin sekalian, sampailah kita pada penghujung acara pembukaan pada pagi hari ini. Terima kasih atas partisipasi hadirin sekalian dalam pembukaan acara ICOLIB kelima tahun 2023. Semoga acara ini menjadi ajang diseminasi ilmu pengetahuan yang produktif dan bermanfaat bagi hadirin sekalian. And now we'll start the first plenary session with 2 speakers. This session will be moderated by Bapak Muhammad Suudi, PhD. Bapak Muhammad Suudi, PhD is one of the lecturer of Biology Department, Faculty of Mathematics and Natural Sciences, and he is experting in plant molecular biology and plant biotechnology. Bapak Suudi will be accompanied by Bapak Arif Muhammad Siddiq, SSI MSI as Notulen.
Bapak dan Ibu sekalian,
Kita mulai jam 9 aja Pak, gak apa-apa. Kan masih ada waktu biar nggak tergesa-gesa. Iya. Oh iya, jadi nanti akan live streaming. Jadi jam 9 kita mulai.
Live streaming, meeting udah.
Iya, jam 9 videonya diputar. Iya, ini sekalian live streaming. Iya, videonya diputar. Oh iya, video tadi Prof Diki. Supaya Pak Sudi sama Diki.
9 jam. 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 9
Terima kasih kepada para hadirin yang telah mengikuti sesi pembukaan acara ICOLIB kelima. Untuk selanjutnya yaitu acara plenary session. Namun sebelum itu acara plenary session akan dimulai pada pukul 9 waktu Indonesia Barat. Maka lebih kurang waktu 15 menit kita akan break sebentar dan sesi plenary akan segera dimulai. Terima kasih.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lecturers, with 14 of the lecturers having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
Sustainable.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student-central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
Sustainable.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
sustainable.
Tes suara.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since
Baik, waktunya sudah jam 9 dan kita mulai lagi conference. Baik, Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, now we enter the plenary session 1, and in this plenary session 1, I will be moderate to I will be accompanied by Mr. Arif, who will be a note taker, and
Once again, on behalf of the committee, I would like to say welcome and warm greeting from University of Jember to all the participants and also distinguished invited speakers in International Conference 5th ICOLIB 2023. Without further ado, now we will start the plenary session 1, which consists of 2 speakers. The first is Dr. Asmoro Lelono, and the second one is Dr. Renat Arif Lukman. For the first lecture, we will give opportunity to Dr. Asmoro in delivering his lecture entitled The Contribution of Avian Research to the Science and Food Safety. But before we focus and pay attention to his lecture, let me introduce him briefly. Dr. Asmoro Lelono now is a lecturer and also a researcher in Department of Biology, Faculty of Mathematics and Natural Science, University of Jember. For the educational backgrounds, he earned bachelor degree from IKIP Bandung, master degree from ITB, and a PhD degree from Groningen University with a specific major behavior ecology. And he also involved in several activities, research, and also community service. And at this time, he is the visiting research fellow in Institute Groningen, University of the Netherlands. And by his research, he already earned several reputable publications, which is published in, for example, like Journal Hormones and Behavior, Scientific Report, Biology Open, and also Journal of Animal Ecology. Ladies and gentlemen, now let's pay our attention to our first lecture from Dr. Asmoro. Dr. Asmoro, the time is yours.
Thank you very much for the chance. So as usually as a lecturer, usually I stand up in the floor. So please let me to do that because it will make me easier to deliver my topic. So as the request of the committee, first I would like to express my honorable feeling that in this occasion I have a chance to share what we did in our group. So for last 10 years, we start to study the avian biology, focus on the endocrinology and also the behavior. So after I finish my PhD, I will start to a new group in a zoology lab. The name is Avian Research Group. So we focus on the study on biology of the avian species, especially for the species that we usually use in a daily activity. For example, the Japanese quail, the Javan ducks, and also the domestic chicken. So in this chance, in ICOLIB 5, I would like to deliver the topic of the contribution of avian research to the science and also the food sovereignty. So first, why it not change the, could you help me please? Okay, yeah. So, because the topic is what the research after the COVID era, it's also become our basic starting point. What happened during the COVID era? So we realized that during that times, the mobility of the people and also the access to the stuff, to the material, to the food would be limited. So you can imagine that people usually go to the market for their food supplies. It's forbidden because the government say, please stay at home. But they should eat every day. And at that time, the food source become more difficult to access. Could you imagine that every day people would consume those protein source, the chicken meat and also the chicken eggs. That's the cheapest food source contain protein. And that's important for the children, for everybody. And at that time, it's become difficult to assess. So it's also contribute almost 10% and also more or less 12 million people depend on this business. So it can imagine that people raise the chicken and also a farmer, distributor, and also the company depend on this industry would suffer because of the COVID era, of the COVID situation. So the next, how many chicken in Indonesia at the moment? More than 5 million. It's only the number of the broiler and also layer, because we could ask to the company how many chicken they deliver every year, for example. But what about the free-ranging chicken? Because many of the villagers have a chicken, have other strain of chicken. We could not count them, but it's significantly contribute to the consumption of the protein itself. So then we consider how we could contribute to this aspect. So the main problem of the poultry production, first, because mainly of chicken meat coming from the broiler production. Broiler mean chicken only produced for meat. It's only take 35 days or maybe less to produce more or less 1.5 kilogram of meat. And one of most important also the layer one. The layer means that the chicken who should produce egg every day. But what's the problem actually with the layer? You know, every time the company who produce layer chick should kill half of the chick. You can imagine half of the chick they should kill. Why? Because it's only female chick available. Male chick is not available, not usable at last. What it means that if the company sell the chick for egg productions, they only concerns with the female one. But because the proportion of male and female is 1 by 1, if they produce, let's say, 500 chicks, half of them should be killed. Because they know there is no need of the male chicks. So you could imagine that, for example, this. For example, in United States, every year they have to kill more than 300 million male chick. So you could imagine how much money they should spend for nothing. So our concerns now, how we could predict or determine the sex of the embryo before they hatch, because it will reduce the number of living male chicks during the production. So that's the first our consideration, because it could reduce the cost. The second one, because most of the costs, no, not most, some of the costs for the egg production and also chick production for meat production is the antibiotic and also medicine to keep the animal healthy. But some of the consumer concern with this kind of a problem, because many of us now concerns that they would avoid any meat productions which contain antibiotic or medicine. So it's also kind of a dilemma for the producer, because in the same times they should ensure that all of the chick healthy, but in the same times they have to use antibiotic and also medicines to do that. In the different sides, the consumer now concerns with the kind of the situation, because they try to avoid anything contained with the antibiotic, because it could be influence their healthy. So that's two of the main points that we try to contribute through our study in avian study. In this case, the poultry one. So it's also considered and also supported by the national research policy, that now we concerns how we could contribute with the production of the food providing by creating immunity strains. What it means, we try to develop a strain of the layer chicken, means chicken for egg productions, which produce more immunity as factor, in this case, immunoglobulin. So at the end, we try to create the chicken which has ability to produce more natural antibody. So without using antibiotic, we try to develop the chicken strain to produce their own antibiotic. That's the same when we happened to us during the COVID pandemic. The government used the vaccine. But in this case, we try to develop the chicken to produce their own antibiotic. So this is my previous study in Groningen. They have a lot. They have annual facility outside. So we try to manipulate or create our small scale of the avian study, avian area for study. So at that moment, I studied two different kind of chicken. First is the red jungle fowl, and the second one is the layer chicken. So red jungle fowl is the ancestor of the domestic chicken. So if you go to near the forest or in the plantation, tea plantation or near the forest, or coffee plantation, if you go to Mount Argopuro or go to Baluran or Alas Purwo, you could see easily this kind of chicken. Yeah, easy to hear but difficult to see. Those are the ancestor of domestic chicken. So the difference with the other one, because we have two different kind of native chicken. First is the green one, Gallus varius, and the second one is the red one. This is Gallus gallus. So we could crossbreed between red jungle fowl with the domestic chicken, produce a fertile offspring. The second one, so you can see in the left one, so those coming from the Wageningen University, they successfully produced two different line of chicken. So first, produce high immunity strain, and the second one is the lowest one. So we try to replicate this kind of chicken through the manipulation of the egg. So we try to manipulate the egg of the chicken to produce both different strain. In Wageningen, they spent almost 15 years to produce different lines. So we try to do that and copy that by a certain kind of method to reduce the length to become 4 or 5 years. I hope that we could achieve that. So what is actually the essential of the method of the sexing? First, the chicken has a different pattern of the sexing. In human, male and female different in chromosome sex. So for the male, XY, and the female, XX. But the chicken are a different one. In human, the sex of the offspring determined by the sperms. Which sperms goes to the ovum? Because sperms contain X or Y. So the determination of the sex depends on the father's side. But in a chicken, determination of the sex depends on the female one, because female chicken or hens produce Z or W chromosome. So she could manipulate which sex or which ovum emerge from the ovarium, and it will become and meet with the sperms with the same Z chromosome. So the determination of the sex would depend on the female side. That's an opportunity for us. Because why? Because we could create which ovum contain Z or W emerge from the ovarium. So the condition of the mother itself, in certain case, could stimulate which chromosome in the ovum emerge from the ovarium. For example, a stressed hen would produce more corticosterone hormone. The corticosterone hormone is the hormone will produce during the stress. So if you make a mother stress, so they will contain a lot of hormone on the inner ovarium, and they will influence only Y ovarium or ovum will emerge and meet with the sperms. So sex bias will happen in a mother in stress condition. So more female than male. So of course this kind of stimulation should we should try to avoid a inner condition in the ovarium. Yeah. So the other problem come from the sexing after egg Yeah. So usually it takes 24 hours from ovulation until egg already produced by the female. So during that time, the female would deposit albumin and cover the eggs with the calcium carbonate. It will become harder. So mother only have a chance to invest anything, nutrient, hormone, antibiotic, and others, only during the 24 hours. That will become arsenal or anything for the egg itself to develop a new individual. Because mother unable to add more after the egg already delivered. So after egg comes out from the mother body, there is no chance for mother to do anything. It's different with the human. Because human has a uterus and mother deposit nutrient and others through the placenta. But the egg, the chicken, unable to do that. That's also the chance for us to manipulate. So during the incubation, there is no chance for the mother to do anything. What the chance of the industrial in this case? So the industrial would care, is it possible to detect the sex of the embryo as soon as possible before the chick hatch? So what we did, actually what they did, they select the sex of the chick by looking the feather. The first day, the female and female already recognized, and then they will select which one is the male, which one is the female. And the female will goes to the box, goes to the farmer, but the male one will goes to the conveyor because there is no value for those. Yeah, they should be killed. So you can imagine 30 million, and the cost of the female chicks already should be covered by this. I mean, if the company sell its chick, a female chick, 10,000 for example, it's coming from 2 individuals because the male one could not be sold. So if we could detect the female one as early as possible, maybe the cost of each female chick could be lower, maybe 6,000 or maybe 5,000. So what we did at the moment, we use Japanese quail, or we call it puyo, because it's a small one, and it only takes 2 weeks for one round. And it's put it into the incubator, and then we try to detect the heartbeat of the embryo. What the idea of using heartbeat? Because the female and male chicken, the adult one, if we measure the heartbeat, the female are faster than male. It means that the metabolism of the female are higher than male. Why? Because she has to produce egg. Male only produce sperms, and it's less. So the female should increase their metabolic rate by pumping blood more faster. So normally male has lower heartbeat. So we try to do that and to analyze whether it's happened also during the embryonic stage. So what we did, we tried to count the average of heartbeat. What we did is we count from day 10 until day 14, because day 15 and 16 they already prepare for hatching in Japanese quail. So you could see that from day 10, 11, 12 until 14, they try to separate. So the female look higher or faster than a male. Yeah. So we then analyze by statistical model using fixed model, and we found a significant difference between male and female. So at day 11, 12, and 14, we could recognize which eggs goes to male and which one goes to the female. The challenge for us is how to apply this in a chicken model. So we have to replicate this many times to come to the conclusion that it could be happen to chicken, and then goes to scale industrial, and it could be economical impact. So we try to do the same in Javan ducks. We try to do the same, but it's still in the process, and also with the chicken. Yeah. So we do it in a Javan quail, Javanese quail at the moment, and then we try to replicate in a chicken, domestic chicken, and also we try to do the same on a Javan duck. that's the first what we did. The second one is we try to develop the resistance strain. In the previous, my previous experience, we use the high and low line of layer chicken from Wageningen University. They use natural selection, means they select which one produce higher and then which one lower, and then put them together, and then we do the natural one. But it will take 15 years to produce. So what we did now, we try to stimulate by injecting the egg, the yolk egg, with a certain stimulation. We could use hormone, such as testosterone, or other substance, to stimulate the production of the immunity. So what we did is we tried to inject the ovum, the yolk one, with the testosterone hormone, and we see whether it changed or could increase the immunity ability of the chicken. So we use a local strain, they called Kuntara. It's more or less similar with the Kedu strain. Yeah. So the appearance are more or less similar with the Kedu. Yeah. So we raised them. So at the moment we use 2 weeks of the embryonic development, and then we inject with the testosterone hormone, and then raise them, and then try to check the cellular immune system. In this case, we analyze the leukocyte, the population of leukocyte, and also differential of leukocyte, because we plan that in the next future we will try to analyze the immunoglobulin. At the moment we decide to use this because it's cheaper. Yeah. For the immunity or immunoglobulin, it costs quite expensive. So now we try to do this first. And if we success, we will replicate this and use immunoglobulin for detection. So we could find some of interesting result. So in the treatment, the leukocyte production is slower. It's also happened with tonic immobility and also in the competitive ability. It means that the control one show better performance than the treatment one. However, in the biomass increase, the treatment one more or less higher than the control one. So we still optimistic that in age of 16 weeks, it could be better performance of the immunity or incompetence of the chick. That's what we did. We tried to replicate some of those. This is the example of what we found now. So we try to create a chicken with the size of the eggs is not small one, but a medium size, because medium size is preferable for the consumption of the children. Because usually, in my case, my son will prefer eat a medium size instead of the biggest one, because it takes a longer time to consume. That's what we already produce now. Yeah, so the recommendation what we could deliver in this occasion, that avian study potential to support food sovereignty because it's contribute of the food source in Indonesian scale, continuing the heartbeat study to industrial scale. So we try to replicate this, the method, hope it could replicate and applicable for the chicken one and industrial scale. So also there are many aspects of the avian biology, especially how to create a immunity line, because immunity line means less use of antibiotic and medicine. It's also easy for the consumer to consume, because they realize or they know that the chicken or any product from the chicken they consume contain less antibiotic and also drug or medicine. That will increase the value of the food itself. So that's my presentation. Hope there is some question from you. Thank you very much.
Thank you very much for very interesting topic and explanation regarding the research on avian field. Now we enter the discussion session, and we have around 15 minutes. So I invite about 3 questioners may have a question. And if the question rise from this room, so just simply raise your hand and the committee will send you a microphone and you can state your question. And if you are in the Zoom, so you also can simply raise your hand and I will notice, and you will also have a chance to ask the question. And please don't forget to state your name and your affiliation. Anybody in this room maybe? Okay, one question. And before I give opportunity to this room, maybe from the Zoom, we may give one questioner. Can we? Okay, not yet. Not yet, maybe, yeah. Okay, I will give first opportunity to Mr. Fuad, please. Maybe committee can help for microphones, so we will, all participants can hear the question.
Thank you very much for the opportunity to give me a question. So I interested about the heat pit rate in the chicken. Based on your experience, have you tried to also observe this pattern in another avian species, for example, bird or another kind of avian species? Thank you very much.
Okay, maybe directly, Dr. Asmara can answer the question.
Yeah, so far we try to analyze or recognize the difference of the heartbeat of the avian species. So basically we could recognize half of the incubation period. For example, if the Japanese quail spend 16 days, so we could start to analyze at day 8. Before that, it will be unable to do that, because the tool will recognize by using the laser system and try to detect the heart of the embryo itself. So if the embryo is still below half of the incubation period, the size is quite small and it's uneasy to do that. So in most of cases, in bird species, the male and the female, as far as I know, the male will be less or slower than a male. So that's actually the normal patterns. We couldn't find any evidence to show a different way around. So far we could do that, but the problem for the scale of industry, how to do as fast as possible and as economic as possible. So that's the challenge. So difference between the research in the lab and also how to apply in industry. But the concept is remain the same. So how we could recognize as soon as possible. And in avian study, avian species, the male less than female. So as simple as that, but to apply industrial scale, It's not that simple. Okay, thank you very much.
Okay, thank you for the answer. Dr. Fuad, is this clear? Or we give one feedback if you still...
Yeah, thank you very much. I want to know if it can be applied to another species, for example, bigger animal compared with avian, for example, a swan or another wild bird. It's possible to use this.
Yeah, it's possible to use, but the problem that we face now at the moment is the size of the tool itself. It's only applicable for the egg, but it's only in a certain size. So we try to do the same to the Javan duck. The size of the egg is bigger. Sometimes you have difficulty to use the tools, but some of the researchers already manipulate the system, and I saw some of our colleagues already use other tools to do that. So for swan and also even for ostrich, it's already possible to do that. Yeah.
Okay, I think that's clear. So I want to move to our participant in the Zoom meeting. Can we take a look? Is there any participant who want to ask the question in Zoom meeting? Maybe not yet, yeah? In the chat column, maybe? Yes. I cannot see from here, actually. Maybe I will go in front. Not yet, okay. So we still have time, about 5 minutes. So actually I have a question, because when I was a student in a biology department, I study about that. For example, like in the reptile. The sex for one individual is really affected by the temperature. So is that possible? We use this kind of approach for the avian study.
Yeah, so the question is, how is it possible to apply sexing in a reptile egg, especially during the incubation period? So as far as I know, that the sex of the reptile will determine in the earlier stage of the incubation period. So for example, the turtle, so the female turtle will digging the sand on the beach and they put the egg and then cover it again. So mother unable to manipulate anything, so she only depend on the weather to incubate the egg to become a new turtle. So in reptile, the sex of the egg will determine at day 5 or day 6. It depends on the temperature of the substrate. So if the substrate is higher from the limit, they will produce more male than female. So in the same times, is the weather or the temperature of the incubation sand, for example, lower than the limit, so the offspring will become more female. So the male and female not depend on the mother, but depend on the environmental condition. But in the chicken, because female already produce Z or W ovum, and it will meet with the Z sperm, so the sex already determined since in the beginning. So there is no chance anymore for us to manipulate the sex of the offspring during the incubation period, because the sex already determined during fertilization. Yeah. So maybe that's the answer to the question.
Okay, thank you very much for the answer. It's very clear. We still have time maybe for one audience who maybe one question again. In this room? Okay. Please state your name and your affiliation.
Thank you so much. My name is Eva Prasetyani Situborang, and I'm a biology student here in biology department. First of all, thank you so much for your very interesting presentation. And my question is very pretty basic. I want to ask, why should we kill the male chicken? You said that the male chicken not economically valued. Why is that? So why can't we just eat chicken? Yeah.
Yes. Okay, please.
Yes, I agree with you. But the problem is that the layer strain will increase the body mass quite slowly. So after a month, male or female layer chicken only reach around 250 gram or 500 maximum. Broiler will able to change the food into the body mass better. So after 1 month, the broiler will increase the body mass not 250 or 500 gram, but almost 2 kilo. So that's why it's not economically anymore. The second one, now already emerged the concerns, especially for animal welfare. Because many people argue, why you have to kill the male one? Because they have a right to live. But the farmers say, who has to pay? Because if we have to raise the male one, we have to spend money, we have to spend a workload, we have to spend anything, and we get enough profit for that. So in certain state of Germany, there is a regulation now that the farmer should raise both male and female, and the government will support, will subsidize. After the male already bigger, then they goes to the slaughter or could be killed. But the cost should be paid by the government because people are already concerned with the animal welfare. They said it's not fair for the male because there is no willing for them to become male, but should be killed because the farmer unwilling to having a male one because they will not produce egg at all. Yeah, that's the question. Thanks.
Yeah, thank you very much for the answer. I guess it's very clear, very different between our humanity and also the company, I guess. Okay, I think a very interesting discussion, but somehow we have a very limited time, and thank you very much again for Dr. Asmoro for delivering the lecture today about avian research. And before we go to the next speaker, please give Dr. Asmoro a big applause. Thank you very much, Dr. Asmoro. Right, now we go to the next speaker. I want to make sure that Dr. Arif already in the Zoom meeting through the virtual from Germany. Yes. Please try one more time, Dr. Arif, your speak.
My voice clear enough or not so clear?
A little bit.
Okay, try to set the audio.
My voice is okay from Germany?
Yeah. Can you
Okay.
hear my voice as well, or not so clear from there?
Yes, clear, already clear. Okay. So we will continue our lecture today. Ladies and gentlemen, now we come to the second lecture, which will be delivered by Dr. Renat Arif Lukman, SSEMT. Now he is a lecturer in Biology Department, Institute Technology 10 November Surabaya, and he has academic career from bachelor degree and master degree in ITS Surabaya, and for the doctor degree he earned from Eberhard Karl Universität Tübingen, Germany, and he has a very reputable experience. For example, like a head of microbiology and biotechnology in Department of Biology ITS Surabaya, and also at the moment he is a postdoctoral researcher in Tübingen, Germany. He also produced one book which is very interesting. Maybe we can also read related with his interest research about bacteria, host interaction, and also microbiome. And several publications, we can take a look in the Google Scholar, and he is very active to write an article, both research and review, and we can just click the barcode if you want to click his article. Okay, that introduction for Dr. Arif. So without further ado, let's pay attention to our second plenary session, which is delivered by Dr. Arif. Dr. Arif, the time is yours.
Yeah, thank you very much, Pak Suudi. Should I share my screen by myself, or maybe the computer could help me to share the screen? Just in case my connection would be not so good.
Yes, if convenient, if you feel convenient, Dr. Arif, you can share by yourself, so you can control. Yes.
Okay, I will try. It's still disabled, the participant screen sharing, so could you please help me for enabling it? Yeah.
Okay, it's fine.
Yeah. Yeah, thank you very much. So first of all, I would like to express my gratitude for the opportunity, for the invitation from the Biology Department of University of Jember, as well as the committee of the ICOLIB conference. And it's still disabled, the screen sharing, yeah. So I will just directly present my research with entitled Unveiling the Intricate Interaction Between the Neurochemical Producing Staphylococci as a Human Commensal with the Host. Could you go please to the next slide? Yeah, so before we go further about the research, I would like to give you some short or brief background regarding the topic. So first of all, about the neurochemicals. What are neurochemicals? Neurochemicals is chemical compounds that related to the neuron activity, for example, which is very well known is the acetylcholine, dopamine, GABA, serotonin, as well as the other neurochemicals. In this case, the examples are tryptamine, tyramine, phenylethylamine, and also dopamine. Yeah, we can go to the next slide, please. And the next term will be about the microbiota in human, and we can call it commensal. What are commensal, or what are human microbiota? As we know that human is a supra-organism which can be inhabited by many microorganisms inside and also outside the body. for example, from the archaea, viruses, parasites, bacteria, and also fungi, which can have a symbiotic interaction with the host, in which, in this case, is a human itself. So we are currently living not by ourselves, but interacting with the other microbiome, for example, in this various domain of the organism. Please go to the next slide. I'm sorry, maybe the slide is not well, yeah. And the next one is about the staphylococci or staphylococcus. As we know that staphylococcus, for example, as aureus, staphylococcus aureus is a pathogenic or dangerous strain that can also make an infection which is deadly and dangerous. And our body can be inhabited by many other staphylococcus, which is not so pathogenic. In this case, we call it the commensal staphylococcus, which from the gram-positive bacteria has a grape-like cluster, and also it can live anaerobically. And some of the species that live in or on our body are Staphylococcus lugdunensis, S. aureus, S. capreae, epidermidis, which can live on the skin of the human, and capitis, hominis, and other species. Please go to the next slide. So the main question for this research is, we have two questions. The first one is, how do staphylococci produce neurochemicals? Because as we know that neurochemicals are only playing in the human body, for example, as in the neuron system or nerve system, while the bacteria has no nervous system. And the second question is, what is the role of this neurochemical producing staphylococci as a human commensal? What do they do in our body? And what is the crucial role of them in and on our body? Please go to the next slide. Yeah, the first, we would like to address the question about how can staphylococci produce neurochemicals? And in this case, we discovered a gene that some staphylococci possess this kind of gene. We call it SAT-A, stands for staphylococcal aromatic amino acid decarboxylase. Yeah. This gene encodes a decarboxylase enzyme with the substrate is aromatic amino acids, and it will convert into the corresponding trace amine. And as you can see here in this HPLC chromatograph, that the wild type has the conversion from the aromatic amino acid into corresponding trace amine. When we deleted this SAT-A gene, there is no more conversion into the trace amine. And this trace amine is, the tyramine, phenethylamine, and tryptamine can also be found in many strains of staphylococci. Can you please go to the next slide? Yeah. So the role of these enzymes is decarboxylating the compounds from the aromatic amino acids into the tryptamine, tyramine, and phenethylamine. This enzyme can also produce or convert the 5-hydroxytryptophan into serotonin, and also the L-dopa into dopamine, which are very well-known neurochemicals in our body. Yeah, can you go next slide, please? Or maybe should I? Yeah, okay. So in this case, in this table, we showed that some of the strain and some of the species can produce trace amine or possess the SAT-A gene. And as you can see here, in the intermediate group, all of them producing the trace amine, while the S. aureus, there is no producer of this trace amine. And in the S. epidermidis, we have three strains. For example, S. epidermidis RP62A, S. epidermidis ATCC12228, and also another one, S. epidermidis O47. Not all of them producing trace amine, but only one of them producing trace amine. It means that this SAD agent is not species dependent, but more to the strain dependent. Yeah. Next slide, please. Oh. I think the data is not really clear in here, but yeah, it should be fine. So we can see in the below graph, this is the focus of this graph, and from this pie chart we can see that 60% of the population, 60% of us are colonized by this trace amine producing staphylococcus in our gut. It means that this is quite prevalent in our gut, and it might also play an important role in the gut of the human. Next slide, please. Yeah. So the next question is, what are they doing inside our gut or inside the human gut? And we checked with the in vitro culture, cell culture, and we showed that in the presence of tryptamine, in here is with the tree, we saw the adherence and internalization. Maybe the slide is not really good. Maybe can you make me able to share the screen so I can really control the slide show, if it's possible?
Okay, I think the committee will help.
Yeah, thank you very much, because maybe the conversion into the PDF is not that good. So yeah. Yeah, now it's much better. Okay, in this PowerPoint or in this slide, you can see that the adherence or the attachment of the bacteria into the gut epithelial cells in this in vitro culture or in vitro assay. I use the gut epithelial cells, and it shows that the increasing of the attachment and also invasion in internalization into the cells in the presence of this tryptamine or the neurochemical stuff. And this is quite interesting because the presence of the production of these neurochemicals can increase, can help the bacteria to colonize our gut. And the next question is, how do they, what is the mechanism behind? Can you go to the next slide, please? Yeah, because the adherence or the attachment of the cells into the gut epithelial, for example, and also the internalization involves the actin reorganization, as you can see here with the pedestal, or providing the cells to attach onto the cells, onto the surface of the cells, as well as engulfing the mechanism engulfment to help the bacteria internalize into the cell. It involves the actin reorganization from G-actin or globular actin into actin filament or F-actin in this case. Yeah, next slide, please. So in this slide, we show that the presence of this neurochemical, the tryptamine, phenylethylamine, tyramine, dopamine, serotonin, increase the CMP level of the in vitro cell culture. And later, we show that there is also increase the F-actin level. And this is, we can see in the picture below. The control, there is a very little green string which represent the F-actin. And in the presence of the tryptamine and serotonin, the number of the green strings are increased. It means that it activates the cell motility or self engulfment to the bacteria into the cells. Next slide, please. So, as you maybe already know, that the adrenergic receptor or the receptor that interact with the adrenaline, there are three types. The first one is alpha-1, the next one is alpha-2, and the last one is the beta. And in this case, these three subtypes of the adrenergic receptor can interact with both epinephrine and norepinephrine and activates them with the different pathway. The alpha one will have a phospholipase C, which later will produce triacylglycerol and connect with the calcium pump, which later increase or decrease the level of the calcium level intracellularly, while the alpha 2 and the beta 1 more into the CAMP level. If the alpha 2 activates, it will decrease the CAMP level, while the beta 2 activation will increase the CAMP level. Yeah, next slide, please. So from the experiment, we can see that with the trace amine or neurochemicals or sub-A expressing staphylococci, which can later convert aromatic amino acids as well as the precursor of serotonin and dopamine into corresponding neurochemicals, it will interact with the adrenergic receptor for the dopamine and trace amines. For the serotonin, it will interact with of course, the serotonin receptor, and later it will decrease the CAMP level because of this interaction. And this decrease of the CAMP level will stimulate the organization of G-actin into F-actin or the filament actin, which later increase the engulfment of the bacteria. So the internalization or the colonization of the bacteria into the cells or into the epithelial cells will be also increased. Yeah, next slide, please. So the next question, because we are finished with the gut microbiota, the next question, whether these staphylococci or neurochemicals producing staphylococci are also present in the human skin. And as we know that from the scientific articles, many publications show that the human skin are inhabited by Staphylococcus epidermidis, Staphylococcus aureus, but most of them are Staphylococcus epidermidis. And whether there are aromatic amino acids on the surface of the skin, and whether there are also adrenergic receptor. And some scientific article reported that these aromatic amino acids are also present because we also secrete the sweat. And the human keratinocyte also has the beta-2 adrenergic receptor, which means that there is possibility that this neurochemical producing staphylococci also play a certain role on the skin of the human. And it will be answered by the next slide. Can you go to the next slide, please? Yeah. So before going further, we performed the screening to quantify how many % of the population are colonized by the neurochemicals producing staphylococci. And from the sampling, we know that around 70% of the population are colonized by the neurochemical producing staphylococci from different species. In this case, there are S. hemolyticus, S. epidermidis, capitis, and also capreae, which most of them, or 88%, are S. epidermidis. Yeah, next slide, please. The next question is, what is the role to the skin? And as we know from the wound healing process, there are steps, for example, the proliferation of the skin, which is important for the healing if we have the wound. And there is also another things, it's called the cell migration, which is done by the keratinocytes. So the cell will also migrate or have the motility to really close the wound area, to make the wound closed, so there is no more infection or there is nothing going into the wound. And from this point of view, we would like to investigate whether this neurochemical has something to do with the wound healing process. Next slide, please. So, yeah, before going with the in vivo experiment, we first performed the in vitro experiment using the keratinocyte cell culture. In this case, you can see in the left side the graph or the photo where we see there is the wound closing or the gap closing between the two tissues. Yeah, can you hear my voice clearly, right? Okay, I will continue.
Yes.
Okay, so the first is the control, where we see from the 0 hour, from the initial point there is some gap between the two tissue of keratinocytes, and after 24 hours, the the cell culture or the keratinocytes will also proliferate as well as migrate into the middle of the cell culture, and it will close the gap in between. In the presence of epinephrine, in this case, this is when we have the wound or we are injured, the epinephrine will increase locally as well as systematically, and in this case, it will inhibit the cell migration. So the closing of the wound will also be inhibited as well. And if we add the neurochemicals that we have or the produced by staphylococci, in this case the tryptamine, phenylethylamine, tyramine, and dopamine, the closing of the gap will get recovered. Although it's not as good as in the control, but the gap closing will be much better if we compare with the epinephrine alone. And in the last panel, we see with the ICI. ICI is the antagonist for the blocker of the beta-adrenergic receptor. This we use as a positive control. And the next, in the right graph, we see the quantification, the percentage of the wound closing. In the presence of epinephrine, the wound closing only 50%, or the motility only 50%, while in the presence of the neurochemicals, the motility will be recovered. It means that the presence of this neurochemical enhances the motility of the keratinocytes. Next slide, please. And the next question is how or what is the mechanism behind? Of course, we go to the mechanism of the beta-adrenergic receptor, as I already stated or mentioned before, that beta-adrenergic receptor also play or has something to do with the CNP level. The activation of this beta-adrenergic receptor in the presence of epinephrine will increase the CNP level. And interestingly, if we add the neurochemicals, the tryptamine, phenylethylamine, tyramine, and also dopamine, will decrease the CNP level inside the cells. So it implies that the presence of these neurochemicals will a little bit antagonize the effect of epinephrine. In this case, we see here the ICI. ICI is as a beta blocker. In the presence of the beta blocker, the CNP level will go to the control level, while in the presence of the neurochemicals, the CNP level will go like half of the level of in the presence of the epinephrine. And after checking the CNP level, we confirm with the F-actin level, which is dealing with the cell migration, as we know that the cell migration has something to do with the reorganization of the cytoskeleton. In this case, it's the F-actin. And in the presence of epinephrine, when we have the injury or when there is a wound, the epinephrine will increase or the present in there, and that's why the F-actin level will decrease. So that's why the cell motility of the keratinocyte will also be inhibited. And in the presence of tryptamine, phenylethylamine, tyramine, and dopamine, this effect of the epinephrine will be inhibited half. So the F-actin level will still be induced, but not as good as in the control level. It means that there is a accelerating effect for these neurochemicals stimulating the cell motility of the keratinocytes. Next slide, please. Yeah, and because there is also another things besides the cell motility, there is a cell proliferation, and cell proliferation has something to do or has correlation with the calcium level. So we also measure the calcium level as well. The high calcium level will increase the cell proliferation, which is important for the wound healing. And in this case, we see here that in the presence of epinephrine, the red line will increase the calcium level. That's why the keratinocyte will proliferate to heal the wound. And interestingly, before we see the presence of the neurochemical will inhibit the CNP or the effect of epinephrine in CNP level, but in here there is no effect on the intracellular calcium level. It means that the presence of this neurochemical will not inhibit the cell proliferation, but only stimulating the cell motility, which is important both for the wound healing. This is the quantification for the below graph, is the quantification of the cell proliferation in the percentage. As you can see here that the epinephrine will increase the cell proliferation, and the presence of the other neurochemical will not inhibit the cell proliferation of the keratinocyte cells. Next slide, please. Yeah. The next one, we performed the in vivo wound healing experiment using the mice model, and in this case we make the wound on the back of the mice. So we shave the back of the mice and then treat it with and make the wound with the diameter around 5 millimeter, and then we treated the wound with the neurochemicals, the tryptamine and others, and as well as the bacteria. And we will observe the diameter every 2 days for 2 weeks. Please go to the next slide. Yeah, so in this slide, we can see first in the control where the wound, the left wound and the right wound, both of them are not treated with anything. So the wound healing acceleration or the wound healing speed are very similar. There's no significant difference. While when we added the tryptamine or the neurochemicals, for example, in this case, is the phenylethylamine, we will see the faster wound healing. So the red one, the red line, is treated with the phenylethylamine around 50 microgram per milliliter, and the black line are the control. And as you can see here, that the red wound, the wound with the red indication, will close faster. From the 8 days, we will see already very small wound because of the faster healing. It means that with the presence of the phenylethylamine, it will increase the wound closing. And the next one, we go to this graph in the top right graph. This is when we added the bacteria supplemented with the phenylalanine. This phenylalanine is the precursor for this phenethylamine. So the bacteria will convert this phenylalanine into phenethylamine. Phenylalanine is the aromatic amino acid, and as you can see here, the trend also similar as we added the phenethylamine. The wound will close faster in the blue line. So the blue line is the wild type where the SAT-A gene, or where there is conversion to neurochemical production. And the red line, there is no conversion into the neurochemical production. It implies that the presence of the bacteria that can produce neurochemical will increase the wound healing in the mouse skin in this case. Yeah, next slide, please. So this is the mechanism or the summary overview. As you can see here, that the neurochemicals are present, or the aromatic amino acid present in the human skin because of the sweating, and this will be used by the bacteria to convert into the neurochemicals, and this neurochemical play a role by accelerating the wound closing in mice model by acting as beta-adrenergic receptor partial antagonist, which later increase the F-actin reorganization. Next slide, please. So the next question is, we have done with the alpha-2 adrenergic receptor as well as the beta-adrenergic receptor. Now we will have something to do with the rest. The last one is the alpha-1 adrenergic receptor, whether this neurochemical also can interact with the alpha-1 adrenergic receptor. And in this case, we use the LN cap cell. This is the cell line that's from the pancreatic cells, which express the alpha-1. And as we can see here in the top, in the above figure, we see that in the presence of epinephrine, the red line, the calcium level will be increased. And when we added the neurochemicals, the tryptamine, phenylethylamine, and the others, there is no increase in the calcium level intracellularly. It means that these neurochemicals will act as antagonists. The effect is to the cell proliferation. As you can see in the graph below, the presence of the epinephrine will increase the cell proliferation, and in the presence of the neurochemicals, together with the epinephrine, it will antagonize the effect of the epinephrine. Therefore, the cell viability or cell proliferation will also be inhibited. Yeah, go to the next slide, please. So to sum everything up, we now know that some staphylococci, which are the commensal of the human gut as well as human skin, express SAT-A gene that encodes the aromatic amino acid decarboxylase, which later produce trisamine, the tryptamine, phenylethylamine, and dopamine, as well as producing serotonin and dopamine. And these SAT-A or neurochemical producing staphylococci are prevalent in both human skin and also human intestine. And these neurochemicals that produced by staphylococci can act differently depends on the receptor. For example, in the alpha-2 adrenergic receptor, it will act as agonist. In beta-adrenergic receptor, it will act as partial antagonist, while in the alpha-1 adrenergic receptor, it will act as antagonist. So this is very interesting with the multi-functional or multi-target or multi-pathway of the interaction, which open a novel interaction pathway between the commensals, particularly the neurochemical producing commensal with the host, in this case, is a human. Go to the next slide, please. So before closing this talk, I would like to acknowledge our gratitude. The first to the Biology Department of Universitas Jember, Indonesia, of course, for the invitation for me to give a talk, and for the Biology Department of ITS as my home base. And the next one is Microbial Genetic Department in University of Tübingen, where I did my PhD. And now the Institute of Molecular Infection Biology in University of Würzburg, where I am doing my postdoctoral fellowship now. And of course to the LPDP for the scholarship for my doctoral studies, and DFG, the Deutsche Forschungsgemeinschaft, for the research funding, and Alexander von Humboldt Stiftung for the scholarship for my postdoctoral. Yeah, thank you very much for your attention. I would be happy for any discussion or any comments and any questions. Thank you. I will give back to the Pak Suud. Thank you.
Okay, thank you very much for Dr. Arif. It's very interesting and nice presentation about the topic of bacterial interaction in gut and also human skin. Now I invite several questions from audience in this room or maybe also in the Zoom virtual meeting. We have about 10 minutes to discuss with Dr. Arif related with his talk. Okay, please. Okay, one from this room, and maybe we also take a look in chat column, maybe. Not yet. Okay, for the first opportunity, I will give to the audience in this room. Please, for committee, could you give microphone so she can state her question. Don't forget to state your name and also affiliation. Okay.
Okay, thank you for the time. My name is Gabriela Integralus from one of the biology students from Jember University, and thank you so much for your amazing presentation. When you explain how the Staphylococcus produce the neurochemical, this makes me have questions about how long it takes for Staphylococcus to produce neurochemical, like does in one mechanism take a long time or more quickly in one mechanism? Thank you.
Okay, Dr. Arif, maybe you can give the answer for the question from our student.
Yeah, thank you, Pak Sudi. So thank you very much for the question. So this is interesting question. The question is how long Staphylococci needs to produce these neurochemicals. So as long as there is the precursor, in this case the aromatic amino acid, the phenylalanine, tryptophan, and also tyrosine, it will be directly absorbed by the bacteria and directly produced the neurochemicals. So the time is very short because it doesn't need any special energy to produce it. So the mechanism is very fast, and as long as the precursors are there, the production will still be going on. Yeah. I hope that's clear enough.
Okay. Is there any feedback from Gabriel?
No, enough for me. Thank you so much.
Okay, thank you very much for Gabriel and also for Dr. Arif for your answer. We still have time, so I invite again maybe a participant from the Zoom virtual meeting. Not yet, maybe. Maybe from our audience in this room. There is one more question from our lecturer. Yes.
Thank you very much for the presentation. So maybe it's already mentioned by you during the presentation, but I still need clarify maybe my question coming from the urgency of the production of neurochemical itself for the streptococcus. Because I still wonder what actually the basic need of the substance for the streptococcus. Do they essential or just kind of metabolic access of the metabolism? Second, and the first, the second one. Do we have or got an impact by the existence of the substance? For example, it will influence our delivery of the impulse in our nerve system, for example, because some of those are comparable with our neurotransmitter. Thank you very much.
Okay, Dr. Arif, you may direct answer the question.
Yeah, thank you very much for the interesting question. So, about the needs of the staphylococci for this conversion, this is still also under investigation. We have no idea why the staphylococci producing these neurochemicals, because all the precursors, all the aromatic amino acid will be directly converted into the neurochemicals, and the neurochemical produced will all be secreted out. There is nothing, no effect on the bacteria itself. We are still investigating this, the effect on the bacteria, not to the host. And as you can see, where I presented my slide, we deleted the gene that has a role in the production of neurochemicals, and the mutants or the deletion mutant has no defect in the growth. It means the growth are still similar compared to the wild type. So we have no idea why or what is the effect of these neurochemicals into the bacteria itself. So this is quite interesting to follow, but we are still investigating it. And the second question about the effect into the nervous system, right? So this is also interesting question, because for the people with Alzheimer will be treated with the L-DOPA. L-DOPA is the precursor for the dopamine. So if the bacteria that produce neurochemicals presents too much, or the abundance is too high in the human gut, it can uptake this L-DOPA and convert it directly into dopamine in the gut. While the L-DOPA uptake for this patient with Alzheimer will purposely, so the purpose of this L-DOPA consumption, if the body will absorb this L-DOPA and in the brain, it will be converted into dopamine. It means that if the abundance of these bacteria are too much in the human gut, this will take over this L-DOPA into dopamine in the gut. So there is very little remaining L-DOPA into the brain that can be converted into dopamine. And for these neurochemicals like the tryptamine, phenylethylamine, tyramine, the phenylethylamine and tyramine cannot go to the brain or to the nervous system because of the blood-brain barrier. But for tryptamine, there is possibility, or I cannot remember whether it's tryptamine or tyramine, one of the neurochemicals can go through the blood-brain barrier. It means that it can somehow affect the nervous system, which is also still interesting to follow. There is still no evidence regarding the effect directly, but there are many papers stated that the gut microbiota can really play a role or can affect the either human behavior or human nervous system indirectly, but the direct effect is still in question. So I hope that's the answer for the question, although there is not so much I can really answer for that.
Okay, thank you very much for Dr. Arif. I guess the question is very interesting, but still under investigation for Dr. Arif research. Okay, there is one last question from the chat columns, so we can take a look in the chat columns. The question is, dear Dr. Arif, very interesting research. One question, did you do histopathology examination on your in vivo experiments? It is important to ensure that the healing was complete until the inner part of skin. Thank you. Yeah. This is from Sri Ningsi. Yeah. Yeah, please,
thank you for
Dr. Arif.
Question. Yeah, thank you, Pak Suudi. Thank you for the question. Unfortunately, we didn't do any histology or anatomically the examination or investigation for this case. We just performed the wound healing closure examination by measuring the wound closure. We didn't do any pathology or histology observation, but this is still currently be done by one of my colleagues in ITS to see whether the healing is really complete, the underneath of the skin, not only the epidermal side, but also the layer below of this epidermal. Thank you.
All right, thank you very much for Dr. Arif. Still, actually, other college, in the ITS still doing this kind of experiments.
Yeah.
Okay, thank you very much, Dr. Arif, for the answer. And although very interesting topic and discussion, but somehow our time is limited. And if all audience here and also participant in the Zoom, if you still have a question, you can write through email, or you can send also in our committee, and we will forward to Dr. Arif. And I believe that Dr. Arif will be very happy to answer your question. Once again, thank you very much for Dr. Arif, and please give applause for him.
Thank you very much.
Okay. The times when we take a look at our time is already 10:30, which is now a break time. And after break about 15 minutes, we here and also all of the participants will be in the parallel session, which is divided into the several rooms. And for this session, me as a moderator and also Dr. Arif as a note taker would like to say thank you very much, and hopefully we will meet in the next plenary session. I give back to our Master of Ceremony. Thank you.
Ladies and gentlemen, Now we come to the break station. For your information, we would like to invite you back at 10:45 to join the parallel session. Thank you.
Hadirin sekalian, saat ini kita akan memasuki sesi istirahat. Dan mohon perhatiannya kepada Bapak Ibu dapat kembali lagi menuju ruang utama pada pukul 10.45 untuk mengikuti sesi paralel yang pertama. Terima kasih.
The Biology Department of University of Jember has offered undergraduate studies since 2001 and has opened the master studies since 2010. The undergraduate study and master study both have accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staff. There are 24 lecturers, with 14 of the lecturers having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student-central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and student associations and extracurricular activities. The students and lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
sustainable.
Nikon's new digital medical device for live on-screen pathology imaging. Streamline your workflow. High-quality images captured quickly and easily. A real solution for digital pathology, Eclipse UI. Observe images in real time. Place the sample with one hand. Wait just 2.5 seconds. Monitor observation for streamlining your workflow. It is no longer necessary to sit for hours looking through microscope eyepieces. The images are shared on screen, suitable for 2 or more people to discuss the samples. The macro image is always on screen to help navigate and find areas of specific interest. Nikon's proprietary imaging technology is also applied. Color reproducibility, after image reduction, and speedy scrolling. Nikon's legendary CF5 Plan Fluor series. Objective lenses are adopted for higher viewability. A real solution for digital pathology. Eclipse UI.
Audio Jungle.
Can you be certain what the future holds? Neither can we. But we know from experience that time brings change and alter questions. Will our world have enough food? Is there a cure for dementia or cancer? What can we learn from the past? Will we take to the stars? And are we even asking the right questions? To find answers, we need development and innovation. And sometimes, the tiniest discoveries and achievements are the things that ultimately have the greatest impact. Still, we don't know what will come. But we know that you will find out. And we will do our best to ensure you can rely on us. Just like for the last 75 years, we are working every day to make your job more efficient. Because only then can we look towards a future that inspires and improves people's living conditions. Together.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master's study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student-central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. we have a biennial science conference called International Conference on Life Science and Biotechnology, to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
sustainable.
Ladies and gentlemen, now we come to the first parallel session. Before the session is started, there will be some information will be delivered by the organizing committee. We hope you enjoy the parallel session.
Hadirin sekalian, saat ini kita akan memasuki sesi paralel yang pertama. Namun sebelum sesi paralel yang pertama dimulai, akan ada beberapa informasi yang akan disampaikan oleh panitia terkait. Setelah sesi paralel yang pertama, dimohon untuk Bapak Ibu kembali lagi ke room utama. Kami selaku MC mengucapkan terima kasih atas perhatian Bapak Ibu dalam sesi pleno yang pertama. Kepada panitia yang bertugas, Bapak Arif Muhammad Siddiq, SSI MSI, dipersilahkan.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master's study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Biology Department of University of Jember supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology, to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
sustainable.
The Biology Department of University of Jember has offered undergraduate study since 2001 and has opened the master study since 2010. The undergraduate study and master study both have the accreditation given from Board of National Accreditation for Higher Education. The vision of undergraduate study is providing an academic excellence in bioscience-oriented and based on the sustainable management of tropical biodiversity. The vision of the master study is to provide an academic excellence in tropical biology concurrent to the science and technology progression. Biology department is handled and coordinated by a head of management department, secretary, and professional staffs. There are 24 lectures, with 14 of the lectures having a doctoral degree, 3 laboratory assistants, and 1 department technician. Department of University of Jember, supported by well-equipped facilities. There are classrooms and laboratories to give students opportunities to learn specified fields in biology. The Biology Department of University of Jember also provides the competency test and license certificate for tissue culture skill. The Biology Department of University of Jember provides the student central and project-based and case learning system in class, and also supports the students to have learning experience outside the class, such as internship programs, exchange programs, independent research studies, research output for the community services, and students' associations and extracurricular activities. The students and the lecturers have the opportunity to write the research paper, and some of the papers are published on national and international journal pages. The department is proud to release and present an own journal page called Life Science and Biotechnology Journal in 2022. We have a biennial science conference called International Conference on Life Science and Biotechnology, to gather experienced scientists in life science to discuss any cases related to life science and biotechnology fields.
Jurusan Biologi Fakultas MIPA Universitas Jember terus meningkatkan mutu pembelajaran, penelitian, dan pengabdian pada masyarakat, sarana dan prasarana, serta memperluas jalinan kerjasama untuk menghasilkan lulusan yang unggul di bidang pengelolaan sumber daya hayati tropis dan berakhlak mulia. Biodiversitas tropis
sustainable. if this works. Do you see a full screen?
Yes, now I can see it.
Very good.
Okay. Okay, then it's set up then.
So you should tell me when you should tell me when I should begin,
Yes.
and then I.
Okay. We will first have to wait the MC will to be handed over to our stage. Ya, Pak Subhanul and Pak Arif, we are still waiting for the MC, right, to start and then to hand over to me, right?
Yes.
Okay. Yes.
Ladies and gentlemen, we are coming to the second plenary session. This session will be moderated by Ibu Dr. Renat Kartika Senjarini. Ibu Dr. Renat Kartika Senjarini is one of the lecturers of the Biology Department, Faculty of Mathematics and Natural Science, University of Jember, Indonesia. Ibu Kartika Senjarini, experting in molecular biology, molecular epidemiology, and vector biology. And also, Ibu Kartika will be accompanied by Bapak Subhanu Waton as the Natolin.
Bapak, Ibu sekalian, dan seluruh peserta yang ada di ruangan Zoom, tibalah kita pada sesi pleno yang kedua. Sesi ini akan dipandu oleh Ibu Dr. Renat Kartika Senjarini. Beliau adalah salah satu dosen di jurusan Biologi Fakultas MIPA Universitas Jember. Bidang ilmu yang didalami oleh beliau adalah biologi molekuler, epidemiologi molekuler, dan vektor biologi. Ibu Kartika juga akan didampingi oleh Bapak Subhanul Waton, SSI MSI, selaku Notulen.
And we would like to hand over this session to the moderator, Ibu Kartika Dr. Renat Kartika Senjarini. To Ibu Dr. Kartika Renat Senjarini, the stage is yours.
Okay, thank you very much, the MC, for giving to me this session. So, am I audible to everybody, Pak Subhanul? Can you listen to my voice clearly before I start this session?
Yes,
Okay. Okay, a very warm welcome to Professor Velavan, who is a very long time good friend of the University of Jember, and who was willing to give his time to be one of the keynote speaker of the 5th ICOLIB at our department, Faculty of Mathematics Science, University of Jember. But before I start this session, let me first give a brief introduction about Professor Dr. Tilummalai Samipi Velavan. He is currently a professor and academic in Germany. We call it Academie Ober Arts. So, yeah, at the Institute of Tropical Medicine, University of Tübingen, Germany. He is also a director and also founder of the Vietnamese-German Center for Medical Research in Hanoi, Vietnam. His research interests are in infectious disease, tropical disease, molecular genetics and genomics, epidemiology, host-pathogen interaction, and diagnostic. Professor Velavan is a senior academic counselor at the University of Tübingen, Germany, and an expert in infectious disease researches. As I mentioned earlier, he is a founder and also the director of PG Care, and currently he is a coordinator of one of the Project Global Health Center funded by the German Academic Exchange Service, DAAD. It is called as a Pan-ASEAN Coalition for Epidemic and Outbreak Preparedness, PACE-UP, which aim to bring together multidisciplinary experts from the South-South region, Asia, Africa, to combat emerging and re-emerging infectious disease. Professor Velavan has trained and mentored many younger generation scientists in low and middle-income countries with a vision to increase, strengthen, and sustain individual and institutional capacity in infectious disease clinical research. He is also the recipient of the 2021 Tropical Medicine Award of the German Society for Tropical Medicine and Global Health. He has published more than 200 peer-reviewed publications, which age index of 40. Currently, Prof. Elafan is a member of Scientific Advisory Board and the review panel of the German Federal Ministry of Health, GHPP-BMG, the German Society for International Cooperation, GIS, the German Academic Exchange Service, DAAD, UNESCO, the World Academy of Sciences, and the National Foundation for Science and Technology Development, and the Ministry of Science and Technology, MOS, Vietnam. He is also active as an editor on the editorial board of Nature Scientific Reports, Tropical Medicine, and International Health, BMC Medical Genomics, and also Pathogens Journal. So, without further ado, I will just hand over this session to Prof. Elafan. You have more or less about 45 minutes, and then which will be followed by a discussion afterwards. So please, Prof. Elafan, time is yours.
Thank you, Kartika. I think I'm audible. Just correct me if I'm wrong. Just give me a heads up if it's okay. Am I audible? Good enough?
Yes, very good.
All right. Time to time, I also receive from the screen that people are in the waiting room. I have to get them admitted, or is someone could take care of that?
Yes, I will take care of that. So you don't have to bother about the participants. Me and the other organizing committee will take care of that. So you will focus on the presentation. Thank you.
All right. All right. Thank you. Thank you, Kartika. Thank you, colleagues from Jember. Thank you, colleagues from Indonesia, for giving me this opportunity. Yet again, with Kartika Sanjurani, who's a very good friend of us. We are working together for the last 3 to 4 years now, and it's expanding more and more in the terms of clinical and epidemiological research in terms of infectious diseases research. So Kartika has asked me to give a lecture on a given topic. I proposed 2 to 3 different topics. Then she chose this one, the Middle East Respiratory Syndrome coronavirus, which I'm trying to give you an overview of what has happened in the last 10 years, because this is exactly 10 years has been completed by 2012, when the very first outbreak was, when the first outbreak was recorded in 2012, and from 2012 to 2022, what has happened thus far in terms of circulation of these viruses in human and animal infections, and what has happened thus far is going to be an overview. Before going to the overview, the talk is structured more or less to give you an overview of all the different types of coronaviruses, including the SARS-CoV-2, etc., 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 but also to give a bit more precise knowledge in terms of one of the coronaviruses, which is the MERS-CoV, or the Middle East Respiratory Syndrome coronaviruses. What made us to get connected in this platform is basically, as Kartika has mentioned, this is because of this particular work that we are working together, which is called PACE-UP. PACE-UP is called Pan-ASEAN Coalition for Epidemic and Outbreak Preparedness. The main objective of this big project, or an undertaking for the next 10 years to 15 years, is to conduct multidisciplinary, operational, basic science, translational, clinical research, clinical trials, implementation research in 2 different situations with a strong South-South cooperation during an outbreak, or how best we could expand and strengthen the capacities that are needed during the inter-epidemic period, which means when there is no epidemic, when there is no outbreak, how best we could strengthen our capacities in terms of laboratory, in terms of academic, in terms of infrastructural and organizational capacities over the period of time. And this program is giving us an opportunity to work more together between South-South cooperation, as Indonesia is one of the partners together with Vietnam. So is the other countries also coming into play in the next 5 years' time. So that therefore we see more and more in-depth collaboration, especially with Indonesia, but also equally with other countries such as Myanmar, Malaysia, and so in the next 5 years' time frame. The program offers you a plentiful of opportunities in terms of exchanges, including there are 12 internships in Tübingen that a particular student from a respective university or a particular staff from a respective university will spend 3 months of his or her time in Germany, in Tübingen, in my laboratory. Subsequently, this particular staff or the student will spend 2 consecutive months in a partner institute back in Africa. So there's a long, strong South-South cooperation which is in place. For instance, we have We have right now from Jember, Nurul is here. There are also colleagues in the past.
I'm sorry.
No, it's fine.
Yes, please continue.
Yeah, good. So there are many of these internships which is happening between Germany and Indonesia, but also you have master thesis in Vietnamese German Center for Medical Research, where any student from any given university from the ASEAN countries are welcome to do their master thesis in Vietnamese German Center for Medical Research for a period of 5 months with a scholarship of 400 euros per month. And for instance, there are also Tiara and other colleagues from Jember are currently doing their master thesis in Vietnam. So there are plentiful of opportunities in terms of not only exchange, but in terms of all different aspects as we speak. And now moving on to a different topic, which is the MERS-CoV. I believe the target audiences are more or less with different scientific background. If I'm right, they are mostly biologists, master's level, bachelor's level.
And also some lectures, but mostly from the biology background.
Okay, great. I believe this is not going to be bombarding with you with too many different aspects, but nevertheless, it gives you a kind of overview, like lessons for both coronaviruses as well. Before I talk more and more deeper into MERS-CoV or the Middle East respiratory coronaviruses, let's talk first about coronaviruses. So coronaviruses is a positive single-stranded RNA virus. It is, as you may know, it is known to infect humans, but it's also equally known to infect other vertebrates, including amphibians, birds, and other mammals. The name corona came from the word Latin. Corona means crown, and it looks like the surface projections like solar corona. So that's how the name coronaviruses came into existence. Of course, the size of the coronavirus, which is an RNA virus, depends between the different viruses. As I'm talking about broader terms in terms of coronavirus, it's somewhere anywhere between 28,000 base pairs to 32,000 base pairs in length. Obviously, like other coronaviruses, including the SARS-CoV-2, which you might have learned in the last 2 to 3 years' time, it always codes for structural genes. And these structural genes, like the SARS coronaviruses, or the nucleocapsid protein, spike protein, membrane protein, and glycoproteins. So I'm talking in principle about any given coronaviruses in a wider picture. Getting back into when was the coronavirus being discovered and how it was discovered. It was discovered in 1960s, as you may see. These are the two main communications. One is by Tyrrell and Binu. The second one is Hamre and Prochnow. So what they did is, as you may see, they collected nasal washings from 21 adults and 2 children shortly after the onset of a flu-like illness. And what they termed this washing is B814. And what they try to do is these washings, which are collected over a period of time longitudinally, has been tried to be cultured in human embryonic tracheal organ cultures. Yeah. Therefore, when they cultured this in the organ culture, they are trying to get an infectious agent. And this infectious agent was yet again inoculated into the nasal cavity of the volunteers, which means, like the Robert Koch postulates, an infectious agent should be able to create an infection. Thus, this nasal washings, which are cultured in the organ cultures, the inoculum is inoculated back in the nasal cavities in human volunteers, where they are able to show that these significant proportion of the subjects were able to get an infection. Equally, it was the same time, Hamre and Prochnow, these are the 2 colleagues, also found that these washings can be grown in tissue cultures from samples obtained from the medical students with colds. And they call this virus which was called as B814 in preliminary by Tyrrell and Binot. Rather, they call this as 229E, basically on the viral culture, what they have grown, but its E is called ether sensitive. OC is called organ culture, whereas E is called ether sensitive. Then they finally made by electron microscopy that this BE814, the infected particles, resembled infectious bronchitis viruses of chickens. This is how the overall coronaviruses were very first discovered from the nasal washings collected from patients with common cold by two different groups, finally with two different etiological agents of coronavirus, one with organ culture, the other one which is ether sensitive, which is called 229E. As we speak about the whole coronaviruses, the coronaviruses are divided into four types. It's alpha, beta, gamma, and delta viruses. I'm not talking here about the coronaviruses of SARS-CoV-2 as a coronaviruses as a whole. So the alpha and beta originate from mammals, especially from the bats, whereas the gamma and delta originate from the pigs as well as from the aves, from the birds. There are 7 subtypes of coronaviruses that can infect humans, which I will talk a bit more in detail. So there are 7 subtypes of coronaviruses that can infect humans. Among the alpha, beta, gamma, and delta, we have the beta coronaviruses known to be the most lethal and cause severe disease and fatalities in the human population. To sum up here, it's about type of the discovery of the virus, but then you have 4 different types of coronaviruses: alpha, beta, gamma, delta. Beta is the most severe that causes the fatalities. As I said, there are 7 different subtypes of coronaviruses, of human coronaviruses. These are the 7 coronaviruses so far that has been discovered and nomenclature has been given. As you see, the first one is the human coronavirus OC43, which was the very first one which was discovered, followed by the HKU1, which is also a beta coronavirus type. Then you have the 229E, which was also from the other colleagues. It's an alpha coronavirus type. Then the other one is the NL63, which is alpha coronaviruses. So these 4 are the alpha and beta coronaviruses. But nevertheless, these 4 coronaviruses continue to circulate in the human population, and it occurs together with seasonal incidents with other viruses as well. Among these 7, there are 3 more, or these 3 coronaviruses are potentially causing severe symptoms, which belong to the beta coronaviruses. The first one being the severe acute respiratory syndrome coronavirus, which was identified in 2003. This is the SARS-CoV-1. And the second one being the Middle East respiratory syndrome-related coronavirus, MERS-CoV, which is also a beta coronavirus, was first identified in 2012. The third one, which is the last outbreak or the recent what we have experienced, is the severe acute respiratory syndrome coronavirus 2, which is the beta coronavirus, which is identified in 2019. If you look into the timeline of coronaviruses, as I said, in 1965-66, the 2 groups have discovered the 2 different types of coronaviruses. One is the OC43, which is the organ culture. Then the second one is the ether sensitive, which is the 229E. Followed by 2003, there was the SARS-CoV-1, which led to 774 deaths with 8,100 patients being infected. Followed by another strain of the coronavirus, which is identified in 2004, called the HCCoV NL63. So NL stands for Netherlands. And in 2005, it's the same, HKU1. HKU is Hong Kong strain, which means identified from a Hong Kong patient. Then you have in 2012, which is the MERS-CoV, 2012, which is the Mediterranean respiratory coronavirus, and in 2019-2020, which is the SARS-CoV-2, which has the highest number of infection and high number of deaths. Although they have high number of deaths with high number of infection, if you look at the case fatality rate, which means how many die of an infection, the highest among the coronaviruses belongs to Mediterranean MERS-CoV viruses. which has a highest fatality rate of 35% in terms of case fatality rate. So these two other viruses are also obviously found again with other respiratory viruses, especially with respiratory syncytial viruses, which means these viruses such as the NL63, HKU1, and 229E and so. Nevertheless, the three major ones which cause the infection is the SARS-CoV-1, the MERS-CoV, and the SARS-CoV-2. Among these three, the highest case fatality rate has been recorded in MERS-CoV. Let's go a bit more into MERS-CoV. So MERS-CoV is a beta coronavirus, as I've said, which causes a viral respiratory disease. The very first case was identified in Jeddah, which is in Saudi Arabia, in the month of June 2012. It was first identified by an Egyptian physician, Ali Muhammad Saki, in a hospital in Jeddah. Equally, the second laboratory proven case was identified also in the same year, but in September 2012 in London. It was then called as London 1 novel COVID 2012, like we call the 2019 novel coronavirus. In those days, in 2012, the second laboratory proven case was called as London 1 novel COVID 2012. The incubation period of this virus is approximately anywhere between 2 and 14 days. The mean is between 5 and 6 days. Obviously, like the other coronaviruses, including the SARS-CoV-2, it has the onset of fever followed by a dry cough, dyspnea, shortness of breath, and it has an accompanied also by diarrhea, which is a typical characteristic feature of the MERS-CoV virus. Pneumonia is common. Viral pneumonia is obviously common, but not that all MERS patients will develop this condition. And MERS-CoV is a zoonotic virus, which means it has a transmission, or the route of transmission is through zoonosis, and they are linked to human infection explicitly in dromedary camels. They are very much abundant in Middle East, Africa, and in South Asia from 2018 onwards. And you know that there are two types of camel populations. One is called Bactrian camels. The other one is called the dromedary camels. This virus is very specific to dromedary camels, not to the Bactrian camels. Bactrian camels has, like the camels has a hump. Like as you see, you have a hump in the camel. So the Bactrian camel will have two humps, which means in the shape of B. So this is not the Bactrian camel. This is one hump, which is the dromedary camel. So the MERS-CoV is the source is bats, and the intermediate host is the dromedary camel, which passes on the infection to the human population. In terms of bats, these are all the Egyptian tom bats from Emballonuridae family, which are also called the sheep-tailed bats, and also simple-nosed bats are also known to be the reservoirs for the MERS-CoV infection. So therefore, there is a zoonotic transmission by coming into close contact with camels, and the person get infected. And if you look into the transmission pattern, most of the transmission happens in three different possibilities. One is in a healthcare associated transmission, which means a patient comes in with a MERS-CoV and he's been admitted in the hospital where you have close contact with the other patients. The transmission rates are much high. And equally, the transmission rates are much high within a household, which means a closer contact within a household gives an opportunity or a window of higher transmission. Whereas when you look at the community transmission, it's not so much as you may see in a healthcare associated transmission or in a household transmission. So this is very much restricted why we do not see much more in terms of community transmission of this virus. Thus, we are able to contain this infection as an epidemic or an endemic to a particular region. How do we define a probable case? A probable case of MERS-CoV is defined by obviously a fever and a pneumonia or an acute respiratory distress syndrome, ARDS, based on clinical or a X-ray or a radiological evidence. And a probable case will be considered if he has one of these symptoms with a history of travel to the Arabian Peninsula in the 14 days time frame, or if he has these symptoms alongside with if this person is being treated in a healthcare facility where in 14 days before the symptom onset where MERS has been identified, or symptoms with a close contact with a confirmed case when the case was ill. So these are the definitions for the MERS-CoV probable patient, which a differential diagnosis has to be done in terms of looking into the diagnostic portfolio for MERS-CoV infection per se. When you look into the MERS situation update in April 2023, there were so far in the last, let's say, 10.5 years, we have 2,604 laboratory confirmed cases with 936 associated deaths, with a case fatality rate of 36%. Nevertheless, if you look at the distribution of these cases, 27 different countries have reported globally the MERS-CoV, and the highest number of cases comes from the Arabian Peninsula and especially from Saudi Arabia, which contributes to the 2,196 cases with 855 related deaths, which even has a higher fatality rate of 39% compared to the global case fatality rate. And when you look into the age distribution, obviously like in SARS-CoV-2, what we have seen, most of the primary cases are more than 50 to 59 years old, who are at the highest risk of acquiring this infection. And if you look at the survival rate, the most of these ones who survive belong to the lower age group, whereas the older age group has higher fatality rate because of other comorbidities which were at the highest risk. In summary, it's an age-dependent distribution of primary and secondary cases of the Mediterranean respiratory coronaviruses, as we have seen in the distribution of the age, as we have seen in the SARS-CoV-2 viruses. So this is a recent data between September 2022 until 12 August 2023. There are 3 cases has been reported to WHO, and 3 of those obviously come from Saudi Arabia, from Makkah, Asar, and Riyadh. If you look at the age group, you see 42, 83, and 85, all are male, and they are non-healthcare workers. But you see there were 2 patients who died, one with 42 years, the other with 85 years old, because the other patient, 42, had a bronchial asthma as a comorbidity, whereas the patient number 3 also had diabetes and hypertension as a comorbidity. And if you see the date of death and date of confirmation, it's almost less than a month that the patient died. I also want to give you a bit of information about the MERS cases, what we have reported thus far in Germany. Germany has reported 3 imported cases returning from 3 different years, one in 2012, 2013, and 2015, of which 1 died and 2 recovered. And these cases are mostly the cases or imported cases which came from Qatar, from Abu Dhabi, from UAE in these different years. This was the very first time in 2015 the MERS outbreak was reported other than the Arabian Peninsula, and importation of the MERS-CoV cases was documented in Republic of Korea in 2015, which led to the largest MERS outbreak outside the Middle East, especially in South Korea. If you look into this population, on September 2018, in 2015, if you see, there are totally 186 laboratory confirmed cases that was thus far reported in the year 2015, which has led to 38 associated deaths because of MERS-CoV-2, which happened in South Korea per se, in Republic of Korea. Yet again in 2018, but then Republic of Korea reported another first imported case in the Middle East, but then it was very well contained, very well responded to the outbreak situation, and thus the community transmission nor the hospital-based transmission did not occur. And if you look, these are the very first reports in 2015 that the MERS-CoV was reported in Asia in 2015, which has the largest number of case fatality, 38 deaths among 186 cases that has been documented. Looking back into the genome of the MERS-CoV virus, as I said, it's approximately like 29,000 to 30,000 base pairs. Like the SARS-CoV virus, SARS-CoV-2 or SARS-CoV-1, or any given coronaviruses, it has the nucleocapsid protein, membrane protein, envelope protein, and so on and so forth. It has a very high proofreading, which has the RNA-dependent RNA polymerases. But what is importantly in particular here is that the receptor binding domain for the MERS-CoV is the host receptor dipeptidyl peptidase, which we call this as DPP4. Whereas if you look back into the SARS-CoV and SARS-CoV-2, this belongs to the ACE2, which is the angiotensin converting enzyme 2, which comes from the host cell, which acts as a binding factor to establish an infection inside a lung alveolar epithelial cell. Whereas in MERS-CoV, the host receptor is the dipeptidyl peptidase 4. An infection has been established. How best you can diagnose this infection? The infection can be diagnosed using molecular tests to diagnose an acute or an active infection, which means the presence of a MERS-CoV virus RNA particle, by a real-time PCR, and the molecular test should be positive by a real-time PCR, not for one particular target of the MERS-CoV, but it should be positive for two specific target genomes or two specific targets within the MERS-CoV genomes. Or it could be positive by real-time PCR for one target, let's say N gene or E gene, but subsequently you should have sequenced the second target. Thus, you can confirm this to be a MERS-CoV infection. So the World Health Organization recommends a testing algorithm to start with an upstream E real-time PCR, and if positive, confirm this with an open reading frame 1A assay or the RDRP or the N gene sequence assay for further confirmation. If both an up E and a secondary assay are positive, then it is said to be a confirmed case. Serological tests such as the ELISAs are not used, and it's not used for diagnostic purpose. It's only used for the surveillance purposes. So diagnostics we have looked into. Let's see what's happening in terms of vaccines. So far, there are plentiful of vaccine candidates which are under phase 1, phase 2, phase 3 trials. As you look into this is the GAMVAC BVRS Combi, which is conducting a phase 1-2 trial with Gamaleya Research Institute. The Gamaleya Research Institute vaccine is similar to the institute which also produced the SARS-CoV-2, the Sputnik vaccine, which is the adenovirus vector-based vaccine. They are the ones who are also involved in the MERS-CoV. The second one is the ChAdOx, as you may know from the name. This is the Oxford unit, which also used this MERS-CoV vaccine candidate, which uses the adenovirus vector-based, which is the ChAdOx MERS spike protein antigen, similar to the one which we had from the SARS-CoV-2, like the AstraZeneca ChAdOx thing. And here they try to use this as a single administration with a homologous prime booster. Currently, there is trials going on with the King Abdullah International Medical Research Center. The third one is the modified Vaccinia Ankara vector-based sequence, which is funded by the CEPI, Coalition for Epidemic Preparedness Initiative, CEPI. And they are doing the phase 1B trials, which is also a German company which uses the modified Vaccinia Ankara virus as a vector to add on to the full-length spike gene of the MERS-CoV with a favorable safety profile. And there's one more which looks at the intranasal vaccine, AVAC 101 from Netherlands, which also goes into trial along with the Coalition for Epidemic Preparedness Initiative, CEPI, where they're trying to look for providing a broader protection against 3 different beta coronaviruses, namely the SARS-CoV-1, the SARS-CoV-2, as well as the MERS-CoV. And there are a few other investigational products which are right now in the preclinical processes. So this is an overview of coronaviruses, but also in terms of MERS. Now let's go a bit more deep into the study, what we have done thus far. So what we know is there is a major knowledge gaps remains how MERS-CoV-2 evolves in terms of lineages, clades, how these variants are emerging in humans and animals. Unlike like in the SARS-CoV-2, we are following up, or surveillance is being done more and more often, but now it's getting more and more less in terms of surveillance. But in terms of last 10 to 11 years time frame, what has thus far happened in terms of the distribution of this virus? Unlike the SARS-CoV-1, the MERS-CoV continue to exist in population, but it's not causing any outbreaks, potential outbreaks, but it continues to circulate and exist in humans as well as in animals. Therefore, we try to do a study in terms of looking at a retrospective study, looking at a 10-year global analysis of the human as well as the camel infections thus far being reported from January 1st, 2012 until the August 3rd, 2022. Considering that the viral lineages evolve over time and space, with certain clades emerging and re-emerging in recent years, what we performed in the study is we collected all the different sequences of the whole genome sequences of the MERS-CoV reported from humans, but also from the animal population to reconstruct how this virus has evolved over a period of time. Kartika, am I audible still? I want to check time to time.
Yes, yes, you still have about 12 minutes. So you are
Okay.
good. I mean, your voice is audible. Great.
All right, good. So what we did is, so we looked into, as I said, we looked for the search terms like MERS-CoV, MERS epidemiology, etiology, dromedary, camels, transmission, bats, virology, diagnosis, nosocomial, hospital-based, and we used the different databases such as the WHO, Food and Agriculture Organization, CDC, Public Health England, ECDC, and Saudi Ministry of Health, all these different med portals and so, to collect and retrieve all the studies, but equally the sequences of the whole genome of the MERS-CoV viruses reported from different animals, but also from different humans, from different countries as well. And we looked at 728 whole length genomes, which were loaded until August 2022. And then we looked at explicitly on the spike protein, which is the predominant protein, which is used for the binding of the host receptor. So the S gene, which is a 4061 base pair, of the 29,000 bases, is in principle being subtracted or looked a bit. Should I continue? It says recording stopped.
Yes, yes, please continue.
Okay.
Yes,
the full
please.
From the full length genome, we just looked into the S gene, 4061 base pairs of the S gene from the 728 full length genomes were extracted, and we looked into how best these clades and lineages and the evolution of this virus has happened in different countries, in different populations, or in different hosts. Before I move into certain terminologies, I would like to give you an overview of two terminologies. One is called lineages, the other is called clade. Some of you may know, some of you may not know. Thus, it's my responsibility to give you a very quick overview what a lineage means and what a clade means, because we are going to use these terminologies in the next few slides for a better understanding. It might be easier for you to get to understand what a lineage and a clade does mean. So lineages are relationship through time between ancestors and descendants. Yeah. Whereas a clade is usually a monophyletic group containing a single ancestor and all descendants. For instance, if you look into lineage, lineage is like the baseline or the generation, yeah, that comes as lineages. Whereas a clade comes or bifurcates from a specific lineage into a clade and thus compromising to different clades. For instance, we are a clade coming out from the ancestor of apes. Yeah, so this is called a clade. If you want to bit more in terms of analogy, I could give you another example. Let's say Mozart had 6 children. None of them had their own children. So which means the composer's lineage has ended after one generation. The lineage has stopped, so the clades are not furthermore. So clade is a branch that includes single common ancestor, yeah, of all of its descendants. I hope you get this right. So let's move into the next slide. So if you look into the results of all the 501 of the 728 sequences thus far has been looked into, the largest contributor of the sequences from the MERS-CoV, obviously from the number of cases, equally from the number of sequences, comes from Saudi Arabia, followed by the United Arab Emirates, Jordan, South Korea, and so. And if you look into the distribution of the pattern in terms of the harboring of these infectious agents, you may see in the right corner from the human population, but also from the different camel dromedarious population has also been recorded in the databases. Also from the bats, also from some clonal population, also from llama, which is similar to the camel population, is also being recorded in Qatar, Jordan, South Korea, and all the different places. Especially from Korea, it's morely only in human population. Whereas if you look into the North Africa, but also in parts of West Africa and East Africa, you also see mostly coming out from the camel population, especially from dromedary and camels. So if you look into the number of genomes that we have extracted per year until 2021, you see the highest came in 2015 and 2016, 2017, but it also reflects basically also during the Hajj pilgrimage. There's a kind of an outbreak which did happen in 2015-16. Thus, we had enough number of cases where number of whole genomes has been deposited thus far. And what we have looked into 501 is 8 of the whole genome sequences of the bats, 226 sequences from the humans, and 265 sequences from the camels has been looked in deeper in terms of the lineages and different clades. The next slide may be a bit chaotic, but I will guide you through. What you see is of these all the 501. So these individual line is an individual sequence of 501 of the different MERS-CoV viruses of the S gene looked into in terms of phylogeny, where you can see there are only 3 main clades, clade A and clade C, and clade B. Whereas lineages, it has from lineage 3, 2, 3, 4, 7, and 5. Clade A and clade B are the oldest clades. They become extinct. Now only the clade B is circulating, which has plentiful of lineages. So each of these lineages are giving arising only to clade B. If you look into the structure or stratification of the different clades, as I said, clade A and clade C have become extinct. As you see, only number of 10 sequences, 2012, and clade 5 is very randomly only from camels in Ethiopia has been reported, but clade B is the most predominant clade, which is currently in circulation, which currently continues to evolve over a period of time in terms of the different lineages which arise, which gives rise to this clade B. And clade B is not only restricted to humans or to camels, but it's existing both in humans as well as in camel population. Equally, when you look into the functional region of the receptor binding domain of the spike protein S1 and S2, it's yet again, you see in terms of the different years, the clade B being the most predominant one, which leads to the lineage 2, 4, 6, 7, as well as the 5, whereas the clade A is non-existent in this whole picture. Clade C is not existent as well in terms of giving arise to any of the given lineages. So any of the lineages do not arise to clade C. So what we could clearly show here is the clade B is predominantly responsible for the lineages which are coming, especially 2, 4, 5, 6, 7. In terms of clades discussion, clade A and C are extinct and are now considered as non-circulating MERS-CoV-2 strains, whereas clade B has evolved further, as you have seen, with multiple viral lineages, which is known to infect both humans as well as the dromedary camels. In 2016, the clade B was described with 5 lineages, but in 2017, within a year time, there are 7 lineages which have been coding for the clade B. So the phylogeographical analysis suggests that the current distribution of MERS-CoV clades in camels is not predominantly shaped by the trans-regional exchange. It appears that despite a steady import of camels from Africa infected with MERS-CoV clade C viruses, the African lineages do not appear to have established themselves in Saudi Arabian camels, which means none of the camels which are even imported from Africa to Middle East are not able to establish a specific MERS-CoV strain in Saudi Arabia, which means the Saudi Arabian MERS-CoV strains are completely phylogenetically distant from the African MERS-CoV-2 strains.
Lavan, I'm sorry, only for reminder, you have only about 3 minutes remaining time.
Oh, that's fine. That's all good. In terms of lineages, we have, as I said, there are 7 different lineages. Most of the 7 lineages belong to the clade B. Among these 7 different lineages, if you look the lineage 4, which is responsible for the Hajj pilgrimage outbreak, when you look at the lineage 5, which is a recombination of lineage 3 and 4, which are responsible for the Saudi Arabian outbreak in 2016 and also in South Korea. And this particular lineage has a higher replicative fitness, which means higher survival and higher transmission rates. Whereas lineage 6 is very unique to human populations, and lineage 7 is also mostly found in dromedary camels. However, there is one patient who was reported from Germany carried the lineage 7. To conclude, the number of human MERS-CoV cases have declined in the past 5 years, but it continued to circulate both in humans, causing sporadic MERS cases in human population. Several new genetic variants of MERS-CoV appear to have evolved from camels and appeared in humans over time. The recombination rates indicate that the co-infection with different MERS-CoV lineages has an adaptive potential in camels. Thus, a continuous surveillance and genomic analysis of MERS-CoV infections, including different variants of concern, is essential and is necessary over time and over space. And MERS-CoV-2 still remains in the WHO blueprint in the list of pathogens considered as a threat to global public health security, requiring more investments for research into surveillance, new diagnostics, specific treatments, as well as vaccines. With this, I would like to leave the floor acknowledging our authors, especially Isam Azhar, Ikromi Rungsung, Tible, David Hui, Brain McCloskey, Sherif, and Professor Sir Ali Zumla. And the article has already been published, which is in June 2023. You are most welcome to read the article in the International Journal of Infectious Diseases. And I will also thank our group, Agi Velavan, in Institute of Tropical Medicine, Tübingen, and equally thanking the organizers and especially Kartika Sinjurani for providing this opportunity to interact and to share my experience and knowledge in terms of the Middle East respiratory syndrome coronaviruses. Thank you so much.
Yeah, thank you very much, Velavan, for very informative and stimulating presentation. You are finished exactly 45 minutes from the time. So for your information, this international conference held in 2 forms. One is in online, as you see here, about more than 90 participants are joining our conference by online, and as you see also about 80 participants also joining offline in our auditorium in Jember. So now I'm going to start the discussion session. So for you, if you have any question, please raise your hand, or you could also write the question in the chat room. I see already one lecture. Any student who is also wanted to ask question and wanted to stay in Bahasa, it is also no problem. Just raise your hand, just ask, and then I will try to translate in English. So the first one was from Pak Siswanto and then Pak Fuad, if I'm not mistaken. Yeah? It was from, Pak Asmoro it was. Sorry. Yeah, Pak Asmoro and then Pak Fuad. Yeah, please, Pak Asmoro maybe first.
Thank you very much for Mr. Velavan with the interesting...
I'm sorry, we cannot listen to your voice.
Okay.
Velavan, can you listen to him? No.
I couldn't listen to him either.
Yeah. Okay. We cannot listen to the voice. Pak Asmoro.
Yeah.
Let's maybe try to say something.
Okay.
I think the operator is still in mute.
Yeah.
It's
still mute.
Oh, is it?
The operator, please.
Yes. Now, maybe try. It was already like something coming out.
Yes, I'm not really sure if my sound could.
Now, yes. Now it is audible.
Okay, thanks.
Yeah. Okay.
Okay, thank you very much for Professor Velavan with the interesting talk. So there are several questions emerged from my mind. First, that the coronavirus already recognized since 1966. So I just wonder how actually change or evolve in the outer membrane of the coronavirus if compared between the first emerged until the recent one. That's the first question. And the second question is the MERS coming from Middle East means the recognition coming from the Middle East region. And the camel is the common animal there. But I still wonder why it's happening in camel and not in goat, cattle, or other animal. Is there any specialty or certain circumstance which allow them to become a host, like, then the interaction with the human more frequent in Middle East? And the third, because it's already outbreak of the coronavirus, so if we compare between the MERS and also the SARS one, I think the more severe is the last one, because it's not only become epidemic but also pandemic. So as a researcher, could you make a prediction? It could be happen maybe in the next future. And if you make a prediction, let's say next 5 years or 6 years, what your scientific base of your prediction for the next outbreak? Thank you very much.
Okay, thank you very much, Pak Asmoro. I think we have already 3 questions. I think it is better for you, Velavan, to answer first from the first questioner, Pak Asmoro.
Honestly, I lost the first question. If I get it right, how did this evolve over a period of time, if I'm right? And the second one, can you repeat the first question? And the second question I can answer. This is regarding why do you see more specific in Middle East? Why you see in camels? Why don't you see other animals also?
Cows, for example. Or
yeah, that's true.
And the other mammalian.
Yeah, because, camels act as an intermediate host. And obviously you have the vectors, which is basically coming from the fruit-eating sheep-tailed bats. And camels act as an intermediate host. The intermediate host has a receptor, which is called the DPP4. The DPP4 is highly expressed in the respiratory system, as well as in the kidney, in the dromedary camels per se. Therefore, they act as a potential receptors. Thus, its infection is partially established. Therefore, it doesn't mean that the camels will have, they will have some symptoms, but they do not have fatalities per se. But then they act as an intermediate host, where people try to have a closer contact with the camels. People also drink camel milk, thus having a very high transmission route for this MERS-CoV viruses. In principle, the answer is DPP4, which is highly expressed in the respiratory system of the camels. Thus, the infection is more predominant in the camel population. There is also equivalent to llama population. There is 1 or 2 cases that's been thus far reported in terms of the camels. This relates to the second question. The third question is, what is your educated guess in terms of when a next outbreak might happen? I mean, the outbreaks are happening everywhere, even now as we speak. But how well it's been contained is the way we see it. As we see in the SARS-CoV, the traveling has been stopped. SARS-CoV-1, I'm talking about 2003, where we could clearly see that the containing the outbreak very early stages is very key in terms of eliminating the viruses. This is what we see in SARS-CoV. Whereas in SARS-CoV-2, things were open, the borders were open, all flights went into China. They came back with Tet holidays, which is the Chinese Lunar New Year holidays. Then an outbreak began and it was an outbreak in January 31st, where WHO said as a public international health concern. 8th of February, it was called to be a pandemic. By March 8th, then it was called to be a pandemic, which is more to the world. When you come to the evolution of the viruses, some of the viruses do not mutate as fast as it could, because it could have a very good proofreading activity because of the RNA-dependent RNA polymerases. Nevertheless, as we see in SARS-CoV-2, even the spike protein, where the receptor protein is there, we have the high mutation rates occurring in the spike protein per se, which led to the new viral patterns such as the alpha, beta, gamma, delta, as well as the Omicron and the Omicron subvariants as we speak. But if you look into the evolutionary patterns of viruses, it has the cycles as you see. There is two stages in viruses. One, either it can be more virulent or it can be highly transmissible. As you may see, the alpha and beta were very highly transmissible. Alpha were especially highly transmissible, but the beta were virulent. Then you had the delta, which was highly virulent. Then came gamma, which is very short-lived in Brazil and so. And then came the Omicron, which was very highly transmissible, but then less virulent. So you have this negative frequency dependent cycles. Some of the rare alleles get fixed, some of the rare alleles get fixed in a population, thus the transmission is occurring, being less virulent, but then these rare alleles become a virulent one at one given stage, so it goes into oscillating cycles per se. So this is one point as we speak. Also there are outbreaks happening over time, because if you look into the databases from international, the outbreak responses, we have like cases of polio 1 and 2 in Nigeria and so, but how well this has been contained is the biggest question. When it comes to the next outbreak, if you think as a pandemic, I don't see that as a potential, but obviously like dengue is there, all through the years. It becomes hollow endemic in areas. It's not endemic, it becomes hollow endemic over seasons. The transmission pattern is there. The genotypes of this dengue serotypes may vary, 1 or 2 or 3 or 4, depending on the geographical distribution, but it does occur in perennial period of time. So, I mean, chikungunya was also there in 2014, 2015, then it was short-lived, then dengue came. Things are obviously happening back and forth, but how best we look into in terms of containing outbreak is the key question and a key response what we put in terms of our effort and capacities. Thus, an elimination can well be reached, and thus an eradication can be achieved at the end. Over.
Yes, Felavan. I think the first question you haven't answered. The first question was about how was the evolution of the, I think it's more into the membrane between the very first emerged into the currently already divided into many strains. I think it was, in this direction. What was happening during those times?
Yeah, the very first case of 2012, as you have seen, the clade A and clade C, which become extinct by now. It's very more specific in those period of time, but then the adaptive potential in camels, because the virus has to evolve, but because the very first cases are very virulent, which means if you look into 8,000 or cases or 2,300 cases from Saudi Arabia, you might see that only we have 35% of case fatality rate, because the earlier cases are very virulent, which means you have relatively high deaths being documented, but nevertheless, there is no any treatment regimen in place and so on and so forth. For MERS-CoV, there was no any vaccine regimens in place, but then the virus has to evolve. It goes into frequency cycles, but also the adaptive potential in camels makes it less virulent or it makes it more virulent, depends on how best they could adapt over the different generations. If there is a recombination even going on within one single cell with two different viruses, then the new virus might evolve.
Okay. So I will, before I move to the next offline question, there is one question online here from Yuda Haryadi. I think this is a lecture from the other department. Is there any chance that virus, SARS or COVID will be disappear or vanish?
Well, that's what we wish, and it appears to reappear. I mean, obviously, the WHO on 31st of May 2023 has downgraded from that to a pandemic to a different different clause. As we speak, it is reappearing in US. It's reappearing also in Europe. It looks like this will be there to continue to live together with us for some time. But how much it may be virulent depends on how good we have been protected in terms of immunity based on the vaccines that we have thus far received from the spike protein of the very old strain, which was even more virulent at a stage when it came as an outbreak. So all the vaccines thus far which we have received, let it be AstraZeneca or mRNA, whatever it is, it's based on the old SARS-CoV. Except for the mRNA vaccine with the BioNTech, we have the Omicron. It's a double booster along with the old strain, along with the Omicron strain. But you can have only antibody responses, but how much of the antibodies you produce against the infected virus depends on how much of your neutralizing antibodies you have to produce. In this proportion of antibodies depends on your host immune system. This is similar like dengue. Dengue is there and then it reappears and then we are getting it again. And how best we are trying to sustain without a vaccine, without a therapeutic measure. This is something appears to be similar in case.
Okay, thank you very much, Velavan. It's getting more interesting. I will hand over the next question for the participant, offline participant. I think it was Pak Fuad, yeah? Yeah, please.
Thank you very much, Bu Kartika, to give me opportunity to ask question to Prof. Velavan. My background is plant sciences. So in the term of evolution of plant, people usually using the hypothesis, the main mutation for plant is from solar radiation. And in terms of virus, which have higher mutation rate, can you explain to us, especially to our undergraduate student, how fast the mutation rate and what is the main factor that's influencing the mutation? And especially, for example, for the SARS-CoV outbreak, we don't really know what is the main factor. It's directly outbreak and become pandemic. But in natural process, can you explain some factor that maybe influence this phenomena? Thank you very much.
Yes, Silvan, please.
Yeah, thank you. I give you a best example is we go into the SARS-CoV-2. We had two of the alpha and the beta during the early stages, as well as the Omicron. Yeah. Imagine how did these variants evolve? Why these variants evolve? Where did it evolve? These variants evolved restrictively in South Africa as well as in Botswana, the Omicron. And how did it evolve? Because these region and these cases were first reported from patients with immune suppression. Because patients with HIV, when they are treated with more and more immune regulatory drugs, these viruses started to evolve over a period of time, which means if you look into one or two studies, I can give you an example. One patient over a period of 85 days, when he is immune suppressed, had 3 different strains within one patient. Yeah, when immune suppression is basically a way for the virus to get evolved in immunosuppressive patients. So if you look into Botswana, as well as from Beta, which is also from South Africa, these are homegrown cases which are arriving from the immune suppressed individuals per se. Then it goes into the community. First is more like cluster transmission, then it goes into the community, but it's always evolved from certain patients with less immune profile per se. The recombination does occurs in these cells. Each, for instance, this one patient had 3 different strains over a period of 90 days. Does this answer your question? Partially?
Yes, Pak. I think it's still mute.
Yeah.
No.
In nature, sometimes we need to know without looking in the several specific people, but for example, in nature, how the mutation mechanism happen. For example, in another kind of virus that's not infected human, they infected animal or they infected plants. What is kind of main factor that's a trigger the mutation? I mean,
the main factor that trigger the mutation, Velavan. The main factor that trigger the mutation.
The trigger is always, we call this as negative frequency dependent selection. The terminology you may read a bit more into, because at one given time point, let's take about a specific gene or an allele. Allele get fixed in a population, and this allele is fixed in a larger population. But then the rare alleles from different population has an added advantage with high virulence. The more the advantage this allele is, the most common alleles start to become rare allele over a period of time. Then the rare allele becomes again to evolve towards a common allele. So this frequency cycles gives a very clear indication in terms of transmission and virulence equilibrium. It means a trade-off between transmission dynamics in terms of higher transmission rates but being less virulent. Or if you look into even in during the COVID-19, if you look into the child population compared to the adult population during the early stages, you see less transmission in children than high transmission in adults. And if you look now, it looks the other way around. There's high transmission in children and less transmission in adult population. So it's oscillating dynamics over a period of time, how the alleles get fixed, how the rare alleles become a virulent one or a transmissible one. But equally, there are many examples, such as the hemoglobin over a period of time, like the even in malaria, sickle cell anemia, and all the different traits. Selection is a biggest name which we could call in terms of terminology, and competition between the different viruses in one single host to evolve. There are plentiful of other host factors. Immune suppression is one thing. Then you have negative frequency dependent selection is the other thing. Then you have a competition between the viruses, which leads to be more virulent or transmission. So a trade-off, this is the other thing. Then you have also the fixation of the allele models in terms of how best I should sustain, rather not to get eradicated or get vanished. So these are the different factors that are known in terms of evolutionary cycles or evolutionary advantages that these viruses try to, because the generation time of these viruses are much more faster, but nevertheless, the occurrence of mutation rates of these viruses depends on the RNA-dependent RNA polymerases. So the high proofreading activity does not allow them to mutate as fast as that of influenza virus, for instance. We have the H5N1, H1N1, like the hemagglutinin neuraminidase keeps changing over a period of time. The epitopes are keep changing over different seasons. Unlike the other influenza viruses, which is also an RNA virus, here it's a COVID, which has a very good proofreading activity. Here the virus evolves for sure, but then in a slower rate because of the RDRP or the recombinant, the RNA-dependent RNA polymerase enzyme activity, what they have. Over to you guys.
Okay, how is it, Pak Fuad? I hope that will answer your question. Yeah, okay. Now I will move to another one question, I think, because the time is already limited. Maybe, operator, can you maybe display the question, the online question it was? Yes. Yeah. Okay, you can read it yourself.
I would like to ask about how do the molecular mechanism differ sepsis pathogens and SARS-CoV-2, how do they affect the clinical outcomes? I'm not sure what exactly the sepsis pathogens that she is talking about. I think it's Tiara, I guess.
Tiara, yes.
Sepsis pathogens can be bacterial pathogens, which can cause septic shock, which could go into the bloodstream, whereas the SARS-CoV-2 is more of the viral diseases, which goes into an upper respiratory tract or a lower respiratory tract. The clinical outcomes does vary in terms of age, comorbidities, as well as underlying conditions, and when they have been diagnosed and admitted and potential treatment is being given. For SARS-CoV-2, we know for sure until last year we do not have any kind of therapy, but now we have Paxlovid. Paxlovid is very good in terms of therapy. Within the very first 2 days, if you take, it helps you in terms of quicker recovery. Whereas for sepsis pathogen, it could be Klebsiella, it could be a bacterial infection or Burkhardt or any kind of pathogens that might cause septic shock. Which can be treated with good antibiotics, provided if you know what is the exact causative agent of this sepsis pathogen. The clinical outcomes does vary between a viral pathogen and a bacterial pathogen that causes sepsis. But obviously for SARS-CoV-2 comorbidities as well as underlying conditions does determine the clinical outcome in patients when they have the clinical course of this infection. Over.
Yeah, I hope that will answer your question, Tiara. And I think we have one more question, Velavan, from the external participants online. This is about the genome of the MERS-CoV, back to the MERS-CoV. So what is the main difference actually between the three clades that you mentioned between MERS clade A, B, and C based on the genome? Yes. And which one is the most dangerous?
There are three clades, as I said, A, B, and C. And A and C are extinct. B is evolving. And B is the one which gives more to lineage 2, 3, 4, 5, 6, and 7. If you look into the clades A and C, these are the oldest clades. C is more existent mostly only in the camel population, whereas A is the oldest or the historic strain, I would say, like the wild type, as we call this, from the SARS-CoV-2. Whereas B is the mutant one, which gives back to plentiful of lineages, and 3 and 4 gave to 5 lineages within 1 year, which means the S gene is evolving with B as a more, it's not dangerous, I would say. It's more evolving within the clade B, giving rise to more lineages. So C is very restricted to camel population, whereas A is more or less the wild type clade, which does not exist now.
Yes. Okay, thank you very much. But I think, as you mentioned, the currently existing MERS-CoV strain is only the B clade, right? Because all the A and C are already extinct. Yeah, I think it's a really interesting session, Velavan. So Professor Velavan will probably come to University of Jember by the end of the year, right? So I think we will have an offline discussion when you are here together with Prof. Dennis Nuryadi from Lubeck. That will be very great. And I think because the time is also so limited, because we have to prepare for another session, I would like to say once again, thank you very much, Prof. Velavan, for coming and joining our international conference. And we are looking very much forward for any collaboration in the future with your institute, and also for our students as well. We have Tiara already finished with the master module, and also we are looking forward also for the next master module for our students. And me as a moderator, for all the participants, thank you very much for your active participation in this session, and I hope to see you again in the next session. I will give over the session to the MC. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum. Thank you very much, Kartika. Thanks a lot. Bye-bye.
Thank you, Velavan.
Ladies and gentlemen, thank you for the participation in the second plenary, plenum. So our next agenda is the parallel session, the second parallel. It will be held at 15 p.m. So every one of you, please come to the parallel session at 3 p.m.
Hadirin sekalian, terima kasih telah mengikuti sesi pleno yang kedua. Acara selanjutnya yaitu acara sesi paralel yang kedua pada pukul 15.00 waktu Indonesia Barat.
Thank you and please come at the parallel session at 3 p.m. Thank you.